So I am Biswanath, and today we are going to revise the whole molecular biology. Okay, the whole molecular biology which is given in your biochemistry. So this is a very important unit, a long unit although. Okay, so I will try to cover the molecular biology in details. Okay, and it will be a very good revision for you. So first, let us talk about basics. So what is RNA? RNA is a single-stranded ribonucleic acid. Okay, and 3' to 5' phosphodiester linkages are present in your RNA. Okay, RNA has 4 bases, that is adenine, guanine, cytosine, and uracil in the place of thymine. Okay, this is replaced by thymine in your DNA. Understood? So these are your nitrogenous bases, and the nitrogenous bases plus your sugar. Okay, this will form a nucleoside, and this nucleoside plus your phosphate, this will form a nucleotide. Okay, so for examples, what are the examples of nucleoside? For adenine we form adenosine, for guanine guanosine. Okay, for cytosine cytidine, uridine. Okay, these are your nucleosides. And nucleotides, you have adenosine monophosphate, guanosine monophosphate, GMP, CMP, your UMP. Okay, UMP. Understood? Then we have some RNA polymerases. RNA polymerases are the enzymes which synthesize the RNA template. Okay, we have 3 RNA polymerases. RNA polymerase 1 in eukaryotes. Okay, note that this is in eukaryotes. We have 3: RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. Understood? So these 3 are your RNA polymerases. RNA polymerase 1 codes for rRNA, this one codes for your mRNA, this one for your tRNA. Okay, and again for rRNA plus your 5.8s rRNA, 5.8s RNA, 18s RNA, and your 28s RNA. Okay, this RNA polymerase 2 codes for mRNA as well as snRNA. Okay, and 3 codes for tRNA and 5s RNA. Okay, some other RNAs are there like snRNA. snRNA is useful in your RNA modification, and your siRNA is useful in RNA silencing. Okay, then microRNA is for the regulation of gene expression. You will see it in later, regulation of gene expression. Okay, and many other RNAs are there. I'm not going to details. Then let's talk about mRNA. About 5 to 10% of total RNAs. Okay, this is called as a coding RNA. Okay, this codes the information for protein synthesis. Like if you have hnRNA is the precursor of mRNA. Okay, this is the precursor. hnRNA is the precursor of mRNA, and in hnRNA if you put RNA polymerase 2, so it will form your mRNA. Okay, this is a mRNA. Okay, then we'll talk about tRNA. What about tRNA? tRNA is also called as an adapter molecule, or a soluble molecule. It is soluble in structure, 15 to 30% of total. Okay, it reads the message of mRNA, So it is just like this structure. The structure is like this. This is the structure of a tRNA. Okay, and then we have a variable arm here, and this ACC sequence is present there in the 3' end. This is a 5' end. Okay, and this loop is your DHU loop. This is your DHU, dihydrouridine loop. Okay, this is your acceptor arm, and this is your T-SIC loop. Okay, this is a variable arm, and this is your anticodon arm. Okay, then we have some unusual bases. So So 3 unusual bases are there, like thymine, your dihydrouracil, okay, and your hypoxanthine. These 3 are your unusual bases, okay. And then you have an unusual nucleotide present here, unusual nucleotide, which is pseudouridine, okay, which is present in your T-cycle loop, okay. Then your rRNA, talking about the rRNA is the site of protein synthesis. It is helpful in your protein synthesis, okay. And it is comprised of 60 to 80% of total, present maximum abundance, okay, max abundance, okay, max abundance. It is also called as a catalytic RNA, okay. The rRNA of prokaryotes and eukaryotes, in prokaryotes and in eukaryotes, let's compare the rRNA. So in prokaryotes we have 70S and in eukaryotes we have 80S, where S is the Spethberg's unit, okay, which is an indirect indicator of the density and size, okay, or it is called as the sedimentation coefficient, okay. The 70S is has two subunits, is 50S as well as the 30S. This 80S has 60S and 40S. If you do arithmetic sum, it is not the arithmetic sum, okay, because the arms are overlapping over each other. And this 50S also has 23S rRNA and 5S rRNA, okay. And this 30S has 16S. This has about 34 proteins and this is about 21 proteins, okay. And the 60S has 28S rRNA, okay, 5.8S rRNA, okay, and 5S rRNA. This has about 50 proteins are present here, and in this 18S rRNA, which has about 35 proteins. Understood? So these are the basics of your rRNA, okay. Then we will talk about the DNA synthesis. Before talking about DNA synthesis, let's talk a little bit about DNA. DNA, you know, there's a right-handed helical structure, okay. The model is given by Watson and Crick. So we are basically discussing the Watson and Crick model. Antiparallel DNA strands are present like this. Okay. This is the 5' this is the 3' end, okay. We have this major groups and this minor groups, okay. Then here bases must be present, about 10 pairs of bases are present over here, okay. The length of this from 1 turn, this one is your 3.4 nanometer, and we have 10 bases here, okay. I have 10 bases here. You know, the purines, adenine and guanine, are called as purine. Pyrimidines are cytosine and thymine, okay. Nucleotides are AMP, GMP, CMP and TMP, okay. You know all this, okay. Let's talk about the DNA replication in your prokaryotes, okay. Talking about DNA replication in prokaryotes, it is bidirectional, okay. The synthesis will occur in 5' to 3' end. It is semi-discontinuous because in one strand there is continuous synthesis on your leading strand, which is called as a leading strand, okay. And there is discontinuous synthesis on your lagging strand, which will generate your Okazaki fragments, okay. It will generate your Okazaki fragments. Understood? And the semi-conservative, you know, semi-discontinuous, as I said, it is also semi-conservative. This semi-conservative nature is proved by the experiments by Michelson and Stahl experiment, okay. Now, who used the heavy isotopes, the N15 isotope, for proving the semi-conservative nature. Semi-conservative means in one strand you have one old strand plus one newly synthesized strand plus one new strand in each DNA strand synthesized in each of the daughter DNA, okay. Then what are the enzymes required for your DNA replication in prokaryotes? So we require DNA A protein, okay. We require helicase, SSB protein, topoisomerase, Topoisomerase 1 and 2. Topoisomerase 2 is also called as a DNA gyrase. Okay, DNA gyrase. There's a clinical associated with it, that DNA gyrase is inhibited by the drug norfloxacin, which inhibits the DNA gyrase. Okay, which is a bactericidal drug, bactericide. Okay, and another enzymes after topoisomerase we use primase. Okay, primase, then DNA polymerase 3, DNAP3. Okay, DNA polymerase 1, and your ligase. These all enzymes are utilized in your DNA replication in prokaryotes. So first, what happens, that in the DNA, okay, a AT rich region is identified, AT rich, okay, which is called as ORIC. This ORIC is recognized by DNA-A. Okay, this DNA-A recognizes the ORIC, okay, and will cause local unwinding. Will occur, and due to this local unwinding, this shape will be formed. Okay, this is called as a replication bubble. So replication bubble is formed. Then after replication bubble, okay, the helicase will act. This helicase will, what it will do, this is the DNA strand, let's say, okay, of one side, this is the DNA strand of another side. So helicase will act like this. This is the helicase enzyme, here the helicase, and this is also the helicase. Okay, the helicase will open up the strands. Okay, this will go like this, this will go like this, and it will open up the strands. Helicase, it will open up strands. Okay, this will open up the strands, and this opened up strands, these have supercoils. So these are stabilized by these proteins over here. Okay, these are proteins, so stabilizing the supercoils. Okay, or the removing the supercoils, not this. Okay, the strands are separated, and these strands have supercoils, okay, which are removed by topoisomerase 1 and 2. Okay, topoisomerase 1 removes from single strand, while 2 removes from the double strand. Okay, and these purple proteins are your SSB proteins, single strand binding proteins. Okay, SSB proteins, and this SSB proteins, this stabilize the unwound strands. Okay, stabilize unwound strands, okay, to release the tension. Okay, to release the tension release. Okay, so the strands are apart now. Okay, they have two type of strands. One is a leading strand, okay, and another one is your lagging strand. Understood? So in leading strand and lagging strand, what changes occur? So leading strand, the synthesis is occurring in the same like in 5' to 3' direction. Okay, synthesis is occurring, and in this also, but this 5' to 3' in our towards the fork. Okay, this occurs towards fork. Okay, but here in lagging strand, it is also occurring in 3' to 5' direction. Okay, but it is away from fork. Okay, and in leading strand you have continuous synthesis. Okay, continuous synthesis, and in lagging strand you have discontinuous synthesis of DNA. Okay, understood? So in discontinuous synthesis, what fragments are generated? We are generating Okazaki fragments. Are generated. Okay, so basically what is happening, that if you draw the DNA like this, okay, let's draw a DNA. Okay, so it's 3' direction, this 5' direction. Okay, and this is 5', this is 3'. So here we have bases also, and here is the replication fork that is generated. This is replication fork that is generated. Okay, so in this here we have the helicase will unwind the DNA. Okay, and this is stabilized, the coils are stabilized by topoisomerases. Okay, and then you may see the SSB proteins attached to here. SSB proteins stabilize the strands. These are SSB proteins. Okay, these are SSB proteins, and let's say this one is the lagging strand. Okay, so synthesis will occur in this manner, one synthesis. Okay, this will just base by base or part by part, they will be synthesized in these fragments called as your Okazaki fragments here. Okay, this is a lagging strand. Okay, and you can observe well that the lagging strand in this also 5-2 direction 3- direction synthesis is occurring, and this in the leading strand your synthesis is continuous. Okay, synthesis is continuously going on. Okay, this is also occurring in a 5-to 3- direction, you can observe, and this is aided by synthesis occurs, and this is aided by primase. Okay, primase. What is primase? Primase is a DNA dependent RNA polymerase. Okay, which synthesizes an RNA primer. Here we have an RNA primer synthesizes RNA primer. Okay, RNA primer, and this RNA primer will continue synthesizing the leading strand. Okay, synthesizing the leading strand. Understood? And this elongation will be catalyzed. The elongation will be catalyzed by DNA polymerase 3. This DNA polymerase 3, what it will do, it will do the elongation. It will form the 3- to 5-. Okay, it has two types of activity. Okay, first of all, it has a polymerase activity, and for proofreading if DNA that synthesis is occurring in properly or not, it has two types of activity. It has also 5- to 3- polymerase activity, and it has 3- to 5- okay, exonuclease activity. This is exonuclease activity is required for the proofreading. Understood? And after this, DNA polymerase 3 is acting. DNA polymerase 1 comes. This is not RNA. DNA polymerase 1, what it does, it removes the RNA primer. Okay, and this RNA primer removing enzyme also has two activities. It has 5- to 3- polymerase activity also. As 5- to 3- polymerase activity, as well as 5- to 3- exonuclease. Okay, the DNA polymerase 3 had the exonuclease from 3- to 5-. It has polymerase 1 has exonuclease activity. Okay, from 5- to 3- exonuclease activity. Okay, after DNA polymerase 1, we will see the action of DNA ligase. DNA ligase is basically a nick sealing enzyme which requires ATP, and it seals the nicks. Okay, seals the nick sealing enzyme, and DNA replication is thus complete. Okay, DNA replication is thus complete. Understood? And the termination of DNA replication, there is a TER-TUS complex. Okay, this is a bit higher in your syllabus. TER-TUS complex, which is resulting in the termination of DNA replication. Okay, this was the story about prokaryotes. Okay, let's talk about the DNA replication in eukaryotes. So in eukaryotes also we have bidirectional replication. Okay, 5- to 3- synthesis. This is also semi-discontinuous, semi-conservative. Okay, semi-discontinuous, semi-conservative. Okay, but the fact that in prokaryotes it has only one ori. Okay, but in eukaryotes you will see many such sequences. Okay, so I was telling that the eukaryotes, okay, they have many sequences. The multiple ori are present. They have multiple ori called as your replicators or your autosomously replicating sequences. Okay, they have multiple ori. So multiple ori are there, so the replication bubble formation in eukaryotes will be also multiple. Okay, like this multiple bubbles will also form. Okay, multiple replication bubbles will form. Okay, so what are the enzymes? First, helicase will be there. Yes, helicase will be there to open up the strands. Okay, then we have a replication protein A which keeps the strands together. Okay, in eukaryotes we have a another enzyme helicase replication protein A, which keeps the strands together, okay, together. Then we can have the topoisomerases, okay, 1 and 2, which will remove the supercoils to unwind the supercoils, okay, and the SSB proteins. The leading strand and lagging strand will be generated in the same manner. Leading strand will show continuous synthesis, and there will be discontinuous synthesis on the lagging strand, okay. The primase, then primase will come. Primase will what it will do, primase will, it's a DNA dependent RNA polymerase, so it will add the synthesize the RNA primer, okay, synthesize the RNA primer, understood. And then afterwards these steps are different, okay. After primase the action is of DNA polymerase alpha, okay. In eukaryotes we have DNA polymerase alpha. DNA polymerase alpha forms a short DNA strand linked to RNA, linked to the RNA primer, okay. It forms a, first it initiates the starting of a DNA synthesis by forming a short DNA strand, okay. Then we can see the action of another thing which is called as PCNA. PCNA is called as proliferating cellular or cell nuclear antigen. Proliferating cell nuclear antigen, what it does, it forms a clamp, okay, forms a clamp for sliding of DNA polymerase, okay. For sliding of DNA polymerase it forms a clamp, okay. Let's say this is the DNA strand, okay. Let's say this is the DNA strand, so this is a clamp that is formed by a PCNA. This is a PCNA clamp, okay. And this PCNA what attaches, okay, then, okay, so PCNA is a strand, okay, but we have to attach it to the DNA. This is attached to the DNA, so this is, if this is the PCNA strand, so we have to open up the strand so as to attach it to the DNA, okay. So opening of the PCNA strand, opening of strand by another factor which is called as replication factor C. What it does, it opens up the PCNA strand. So PCNA strand is now open and it can now attach to the DNA, okay. And this PCNA and replication factor C, they will form the basis of attachment of the DNA polymerase, okay, the DNA polymerase. And to the leading strand, the DNA polymerase epsilon is attached. This is the delta epsilon. And to the lagging strand, okay, the DNA polymerase delta is attached, okay. This is to the lagging strand, okay. Afterwards, after synthesis of the DNA, we have to remove the RNA primer. So RNase H1 is used to remove the RNA primer, okay. And then but the last nucleotide of the RNA primer, this is retained. So afterwards we will see the use of FEN, flap endonuclease 1, which will removes last nucleotide of RNA primer, okay. This will remove the last nucleotide of your RNA primer. And then we'll see the action of your DNA ligase, which will seal the nick, okay, nick sealing. So a bit complicated than your prokaryotic replication. So what are the requirements? So we need also energy for your RNA primer synthesis, okay. For RNA primer synthesis we need this ATP. This primase will need ATP, okay, ATP, GTP, okay. This is energy is derived from ATP, GTP, CTP, UTP, okay. This is all understood. And for synthesis also we need DATP, DGTP, DTTP and all, okay. Done. So you can compare the RNA polymerases in prokaryotes and eukaryotes also. Prokaryotes have 3 RNA polymerases, 1, 2 and 3. Okay, in which I am talking about DNA polymerases. Prokaryotes have 1, 2 and 3. Okay, and the DNA polymerase 3 in prokaryotes, the highest polymerization rate, okay, about 250 to 1000 nucleotides per second. Okay, and in eukaryotes we have alpha, beta, gamma, delta and epsilon. Okay, alpha is what is alpha doing? It is synthesizing a short stretch of DNA. Beta, DNA polymerase beta, this is useful in your DNA repair mechanisms. It is useful in your DNA repair. Okay, DNA polymerase beta is useful in your DNA repair mechanisms. Okay, and gamma is used for DNA polymerase gamma. This are not yet discussed, so I am discussing it here. This is for mitochondrial DNA synthesis, empty DNA synthesis. Okay, DNA polymerase gamma in your empty DNA synthesis. And epsilon and delta and epsilon, you know, this delta will act as polymerase in your lagging strand, and this epsilon will act as polymerase in your leading strand. Understood? Leading strand, lagging strand polymerizations. Understood? So this completes your DNA and RNA synthesis. Okay, let's talk about another topic which is called as your DNA damage. Okay, let's talk about your DNA damage and how what are the repair mechanisms for DNA damage. Okay, so repair is critical for maintaining the genomic integrity. Okay, and if there is a DNA damage, then there be mutations. Okay, so mutations must be prevented from transmitting to the next generation. Okay, must be prevented from transmitting to the next generation. Okay, so DNA damage, about your DNA damage, what are the damage causing agents? So free radicals, you have physical agents like your UV rays, your X-rays. Okay, this all can cause damage. And chemical agents like your aflatoxins. Okay, your alkylating agents, your hydrazine, ozone. Okay, ozone. These all are chemical agents which will cause your DNA damage. So types of DNA damage. If you want to know the types of DNA damage, so you can have basically 4 types of single strand alterations or single base alteration, your 2 base alternation, your chain breaks and cross linkages. Okay, Okay, so we have single base alteration. Okay, 1 base is altered. Single base alteration. You can have deamination, depurination. Okay, deamination is there. Depurination. Okay, your deletion, your insertion. Okay, these all are single base. And we have 2 base alterations. Okay, 2 base alterations, like your TT dimer formation. You have TT dimer formation, your alkylating agents. This will cause your 2 base alterations. Then there will be chain breaks. Okay, the DNA strands are broken at different points. Chain breaks. Okay, this occur due to your UV radiations, your X-rays. Okay, your gamma rays. Okay, your radioactive disintegration. Understood? And then you can have cross linkages. Okay, which may form between the bases of opposite strand, or same or the opposite strand, or between the DNA and the proteins. Okay, so we have to repair this DNA damages. So we can have repair mechanisms for this. Okay, let's talk about the DNA repair. Talking about DNA repair, first one is the mismatch repair. Okay, mismatch repair. Let's first what it happens that we have to, this is helpful in your single base alterations. alterations, okay, to repair single base alterations. So let's say this is our DNA strand, we have T, and this base has to be removed, this cytosine has to be removed in place of this, okay. This C has to be removed. As we know, T is paired with A, okay, understood. So we have to remove the cytosine. So what happens first is that there will be let's say there is a sequence CTAG here, okay. Then there will be methylation by methylase, okay. There will be methylase activity. Methylase will add the this methyl group here, okay. After methyl group, this will be recognized by MLH and MSH, okay, recognized by MLH and MSH, okay. MLH and MSH will recognize this, okay, this mismatched base, okay. So in this you can see like this, that T and C are there, and the MLH and the alpha MSH, this will recognize the region by the CS3 tag, okay. After recognition, what it does, that there will be exonuclease activity will occur. Exonuclease activity will remove the problematic base, okay. T will be retained but C is removed, okay, C is removed, understood. Then after, there will be DNA polymerization, and DNA polymerase will act. DNA polymerase will synthesize the new base, okay. This will synthesize the new base, this T will be there, and the new base A will be synthesized here, okay, and the CS3 will get retained, this CS3 will get retained, okay. Then after DNA polymerase, we can have DNA ligase, okay. DNA ligase will just join the strand, okay. The nick will be sealed, the T and E. This is our correct combination, okay. This is called as a mismatch repair. And if there is faulty mismatch repair, okay, faulty mismatch repair, then it is called as your hereditary non-polyposis colon cancer, hereditary non-polyposis colon cancer, okay, will cause, okay, colon cancer will cause NNPPC. This is also called as a Lynch syndrome, okay, Lynch syndrome, in which there is a mutation in MLH and alpha MSH. This inherited as a autosomal dominant condition, autosomal dominant inheritance, okay, understood. Then after the mismatch repair, we can have second type of repair, which is called as your base excision repair, okay. In base excision repair, this is caused to treat the damages caused by your deamination, okay. For deamination, like the cytosine is converted into uracil, okay. This is called as a deamination. The amine group of cytosine is minus is there, okay, and uracil is found, okay. Deamination or your depurination, okay, like your GC was there, in this was correct, and after there is deamination, okay. So C is converted to your U, so G will be there and U will be there, okay. This is your DNA, this is a faulty DNA, this is faulty, okay. So what happens, we have the base excision repair, okay. So we basically, what we do is that there is an enzyme first which will cause uracil DNA glycosidase. This uracil DNA glycosidase, it will recognize this U, this is recognize this U. So first step is a recognition, and then it will cut it, okay. It will remove it by generating a basic site. This A basic site is generated, okay. This is glycosidase enzyme, it will cut the base from here and generate an A basic site, okay. Then there will be total removal of this, okay. There will be removal, okay, by apurinic or apyrimidinic exonuclease, AP endonuclease, will remove this, okay. There will be removal, okay. 2 is your removal, Okay, one is the recognition, then we have the removal, and after removal there will be synthesis by DNA polymerase. Okay, DNA polymerase will synthesize, and afterwards the ligase will act and your correct base will be formed. So just we are removing the base, and what are the enzymes? Uracil DNA glycosidase and your epurinic or epyrimidinic endonuclease. Okay, understood? This is a base excision repair. Let's talk about your nucleotide excision repair. The third one is your nucleotide excision repair. The nucleotide excision repair, this is induced by UV light. UV light, that means the damage is caused by the UV light will be repaired by your nucleotide excision repair. Okay, you know UV light will form TT dimers. Okay, this will form TT dimers, so we have to remove the TT dimers. Okay, so to remove the TT dimers we can do like this, that let's take a DNA strand with TT dimers. Okay, here you have TT dimers. Okay, so there is the recognition first, that is the recognition. Okay, as the recognition, this TT dimers, the recognition, and after this in this recognition what happens, that an enzyme called as exonuclease, not exonuclease, exonuclease binds to dimer. Okay, this exonuclease will binds to dimer. Let's say this is the exonuclease which is binding to dimer. Okay, and this will remove the affected segment. Okay, this will remove the affected segment. Okay, so the affected segment is removed. So second one is the removal of segment. Okay, then afterwards there will be DNA polymerase acting upon it, DNA polymerase here, and afterwards there will be DNA ligase acting upon here, DNA ligase. Okay, and DNA ligase will synthesize the DNA again. Okay, so this is called your nucleotide excision repair. Okay, and just know for your information that your TT dimers, this TT dimers can cause your sensitivity, photosensitivity. This can cause sensitivity to your sunlight. Okay, this can cause your sensitivity to your sunlight, and you can have freckles on the skin, freckles on skin. Okay, freckles on skin. And you have, if you have the faulty nucleotide excision repair, faulty nucleotide excision repair, then you will suffer from a condition which is called as xeroderma pigmentosum. Understood? Xeroderma pigmentosum. Afterwards you have double strand break repair. Okay, so you have 4 double strand break repair. Okay, double strand break repair. So your both the strands are broken now, and this is caused due to your ionizing radiations. Okay, your oxidative stress, and the bleomycin drug, which will break both the strands of DNA like this. Okay, so you have first recognition. For recognition we have special type of proteins which you known as Ku proteins. Okay, Ku proteins. So what will the Ku proteins do? That if this is a DNA strand, they will bind to your this part. Okay, the Ku proteins will bind to it, and due to the binding, okay, an enzyme will come, that is your DNA dependent protein kinase. Okay, so there will be first, and then approximation by DNA dependent protein kinase enzyme, okay. So it will, what it will do, it will bring the strands closer. It is called as a approximation, and due to this approximation, what and there is activity by the helicase enzyme. Okay, helicase which will break the strands in this manner, generating some sticky ends. Okay, you generate some ends which can bind to one another. Okay, so here one end is generated, here another end is generated. Okay, this can connect to one another. Okay, so there is base pairing, and excess nucleotide is removed. Then you can have DNA ligase, and DNA ligase will seal the nick, and this your whole DNA strand will be formed. Okay, and your faulty double strand break repair, this is faulty, this is faulty. Okay, you can have disorders like ataxia, telangiectasia. Okay, then you have Fanconi's anemia, okay, Fanconi's anemia, and you can have Bloom's syndrome also, Bloom's syndrome. Understood? And at last you have another DNA repair mechanism which is called as your direct repair. In this direct repair mechanism, you can see the formation in the cases of direct repair is involved in the cases of if pyrimidine dimer is formed. Okay, pyrimidine dimer formation or alkylating agents or modified bases if the bases are modified. Okay, like in the place of guanine you have. Okay, so in the presence of modified bases for you can have examples of modified bases like your guanine, which is converted into your methyl guanine. Okay, methyl guanine, for example, your methyl guanine. Okay, so this methyl guanine, the direct repair does is that we have methyl transferase enzyme. Okay, this methyl transferase which is active form of methyl transferase, it will remove, okay, it will transfer the methyl group and convert itself into a methyl transferase inactive form, and this methyl guanine will again be converted into a guanine. Okay, okay, so this is called as your direct repair. So here we complete our DNA damages. Okay, talking about another thing which is called as your telomeres, a small concept which is called your telomeres. Telomeres are short TG rich repeats. Okay, it is present at the end of the chromosome. Okay, it has a sequence TTAGGG. Okay, this is 6 nucleotide sequence. Connect 5' and 3' end here. Okay, this is the telomere. Okay, and it has around 1500 base pairs and is maintained by the enzyme telomerase. Okay, so what are the functions of telomeres? So telomeres have many functions, like it corrects the replication errors. Okay, corrects replication errors. Okay, it prevents loss of DNA. Okay, DNA molecules loss is prevented, and prevents fusion of your DNA molecules. Okay, but DNA strands can fuse due to approximity, so it prevents the fusion. Okay, and it protects from nuclease activity. Okay, nuclease activity, and gives stability also. Okay, so it is absent in your somatic cells. It is not present in your somatic cells, but it is present in your germ cell and your stem cell. Okay, about nucleus, another thing, that aging is inversely proportional to the telomerase activity. So if there is more aging, then the telomerase activity decreases. Okay, if there is aging, the telomerase activity decreases. Okay, understood? So this is mainly about your histones, about your telomeres. Okay, then we will move on to our core topics. That is, first of all, we will see about transcription. Okay, you know the central dogma. The central dogma is like this: central dogma of molecular biology. Okay, you know the central dogma. From DNA, your RNA is formed, and from RNA, your proteins are formed. Okay, this is the central dogma of molecular biology. Okay, then we have a reverse central dogma, or it is called as teminism. Okay, it is from DNA, RNA is formed. From RNA again DNA is formed. Okay, sorry, I told it in a wrong way. Okay, this is a advanced concept. From RNA, your DNA is formed, and from DNA again RNA will be formed, and from RNA the proteins will be formed. Okay, and the enzyme here from RNA synthesizing from RNA DNA synthesis, this is catalyzed by a reverse transcriptase. Okay, reverse transcriptase. So this is called as teminism, and it's seen in HIV viruses and all. Okay. This is your teminism. Central dogma and teminism are done. Okay, let's discuss about transcription in your prokaryotes as well as transcription in your eukaryotes. Okay, so we'll discuss about transcription and your post transcriptional modifications. Okay, so let's discuss about transcription. So you know transcription, this process, this DNA to RNA conversion, this is called your transcription, and this is called as a translation from RNA to proteins. You know this transcription. Let's start about transcription. Okay, here the main enzyme is your RNA polymerase. Okay, in prokaryotes we are talking about transcription in prokaryotes. Okay, then we'll talk about the eukaryotes. So RNA polymerase in prokaryotes, this is composed of the structure of RNA polymerase in prokaryotes is somewhat like this. Okay, this sigma cofactor is there, two alpha subunits are there. Okay, a beta subunit, a beta dash is there, and a epsilon subunit is there. Okay, so you have a cofactor here, and a core enzyme here. Here is the core enzyme. Okay, the coenzyme and the cofactor here. This cofactor is called as a sigma factor. This is a sigma factor, is the cofactor. This core enzyme is beta dash alpha omega. Okay, this is written in this fact. Okay, this is the cofactor plus your core enzyme makes your holoenzyme. What are the functions? So sigma factor, first of all, sigma factor scans the DNA to recognize the promoter region. So in transcription, the transcription starting is from a promoter region, so it recognizes promoter region. Okay, it recognizes the promoter region, the sigma factor. The alpha subunit, it recognizes the DNA to be transcribed into your RNA. It recognizes the DNA to be transcribed. Understood? And this beta subunit, this beta has a polymerase activity. This beta dash has an helicase activity. This epsilon, the function of epsilon or your omega subunit or epsilon subunit is somewhat ambiguous, so I am not stating this. Okay, this RNA polymerase in prokaryotes, it lacks specificity. Just importantly remember this fact, it lacks specificity. Okay, it lacks specificity, but in eukaryotes RNA polymerases, okay, they have specificity. They have 1, 2 and 3. Okay, so we'll read about them. Okay, so let's draw the DNA and what are the regions of DNA and how it is related with your transcription process. Okay, so if it is the DNA, okay, then we have a +1 region. This is the coding strand. This is the coding strand, and this is a non-coding strand. Okay, so we have a +1 site, this is called transcription start site, TSS. Okay, and at -10 TSS, and at -10 position we have a box, okay, which is TAAT. Okay, if we go in this side from to the left side, okay, from the +1 region, it is called as upstream. Okay, upstream, and if you go downwards, then it is called as a downstream. Understood? And upstream is negative and downstream we take the sign as positive arbitrarily. Okay, so in -10 region we have a TAAT box. Okay, this is called as your Tata box or your Pribno box, or your promoter element. Okay, okay, then you have another box in this region, -35 region. Okay, -35 coding region, TTGACA. Okay, this is a consensus sequence. Okay, this is the 5' TTGACA 3'. Okay, this is the initial point of contact of the holoenzyme. So what happens first? Okay, let me explain the transcription process in this. Okay, so first there is the action of a sigma factor. So sigma factor scans the DNA and binds to the consensus sequence first. So let me draw a DNA and explain to you. So let's draw it. Okay, so here I said it is the -35 region and it's the -10 region. Okay, this is the +1 transcription start site. Okay, so first what happens, that the sigma factor, it scans the DNA and binds to here the -35. Okay, and along with it serves the binding of the core enzyme, the CE. I am writing CE, core enzyme. Okay, and it forms a closed complex here. This is called as the formation of closed complex. Formation of closed complex. Then the holoenzyme slides this whole part, it slides here. Okay, it slides here and to the next sequence, it is called Tata box, TATTAAC box. Okay, TAAT. Okay, the Tata box. And as it slides, okay, as it slides to -10, as it slides to the Tata box. Okay, this whole part, this slides to the Tata box. Okay, then the DNA strand here start unwinding. Okay, and here at the Tata box, okay, there is the formation of an complex which this unwinding goes here and forms a open complex. How the open complex is formed, let me tell you that due to the melting, this Tata box is a site of melting. Okay, this Tata box is a site of melting, or melting means unwinding of DNA strands. About 14 base pairs of DNA get unwind here. Okay, and such type of structure is formed. This type of structure is formed here. Okay, this is called as your transcription bubble. Okay, so transcription bubble formation is seen in this region. Okay, transcription bubble formation is seen in this region. Okay, and the closed complex is formed. Okay, so transcription bubble is formed. Then to this transcription bubble, okay, and the RNA polymerase get attached. Okay, here is the RNA polymerase. It is still attached to the transcription bubble, and in this a stretch of 5 to 10 nucleotides are formed. Okay, this is 1, this is 2, this is 3. Okay, formation of 5 to 10 nucleotides RNA. Okay, which occurs in 5' to 3' direction. Okay, and this RNA polymerase, it moves a bit away from your initiation site. Okay, moves away, which is called as a promoter clearance. Okay, this is called as a promoter clearance. Understood? So promoter clearance is there. So this is the whole step of the initiation. Okay, this is the initiation step. Then along with, we can have termination steps, not the termination, the elongation steps. Okay, so second one is the elongation. Okay, in elongation step, what happens, that you have the DNA like this. Okay, this open transcription bubble is there. So you have the RNA polymerase in this region, and the RNA polymerase will synthesize the template RNA strand. Okay, a transcript of the DNA segment. Okay, so it starts synthesizing a transcript, and the sigma factor is released. Now. Okay, and the core enzyme moves along the template. Moves along the template. Okay, and the transcript is formed. The transcript starts forming. Okay, and how the termination occurs. Okay, termination. So we can have about two theories for termination. We can have either intrinsic termination, or you can have your row dependent termination. So intrinsic termination means you have your process going on here. But what happens is that a series of your C and G. Okay, as G and C have the tendency to be attracted, they bind. So due to this sequences, many G's and C's, they come and form a hairpin loop type structure. Okay, a hairpin loop is formed, C G C G C G G C G C C G. Okay, this loop like structure is formed, which is called as the GC stem, and due to this hairpin loop, okay, due to this the process is blocked, or here you can have a string of use. Okay, a string of use at 3 dash end, this is 3 dash end, a string of use, okay, use, which will bind with A's and block it. Okay, so there are intrinsic signals like a string of use or your GC stem formation, which will terminate the transcription process, or you can have a row dependent termination where a row protein is used. Okay, row protein is used, and row protein is a rotating protein. It is a rotating protein. Okay, rotating protein. Actually, row protein is a RNA stimulated ATP dependent helicase protein. This is a RNA stimulated ATP dependent helicase. Okay, this is a RNA stimulated ATP dependent helicase protein. Okay, so you can have row factor or your row protein. Okay, which is a hexameric protein, also hexameric. Okay, and how it stops that, it binds to your 5 towards your 5 dash end. Okay, and blocks that formation of your transcript now. Okay, generally it is a hexameric it binds to here. Okay, and when it reaches here, it blocks the process here. Okay, the transcription is blocked here. Okay, and about the clinical thing, about the clinicals. Okay, two clinicals are there. Here we have rifampicin. Okay, rifampicin or rifampin. Okay, this inhibits the transcription. Transcription by binding to beta, binds to beta. Okay, binds to beta subunit. Okay, this terminates the chain growth, because beta is helping in your it has polymerase activity, so it is blocking the polymerase activity. And rifampicin is an anti-TB drug, anti-tubercular drug also. Rifampicin is an anti-tubercular drug. Okay, and rifampicin is also helpful in your leprosy also. Okay, leprosy also. It is also bactericidal for Mycobacterium leprae as well as Mycobacterium tuberculosis. Okay, and afterwards we can have another clinical, which is your drug dactinomycin or your actinomycin D. Okay, actinomycin D, which is basically an antibiotic, and this antibiotic intercalates between DNA bases, between DNA bases and inhibits transcription initiation. Okay, it is inhibited from the initiation. Okay, understood. Now let's discuss the transcription process in your eukaryotes. Okay, in eukaryotes it's a bit complicated and involves many factors. Okay, many factors are used. Okay, this was the transcription process in your prokaryotes. Okay, the Tata box, and how the whole enzyme slides in transcription bubble formation, how the promoter clearance is there, nucleotide formation is there. Okay, elongation, how the sigma factor is released. Okay, termination, intrinsic and row dependent. Okay, and clinical we have rifampicin and your dactinomycin. Okay, let's talk about your transcription in your eukaryotes. Okay, talking about transcription in eukaryotes, it is more complicated than prokaryotes. Okay, the start where the transcription starts, it is called as a fidelity. Okay, so fidelity is the start site for transcription in eukaryotes, the +1 region. Okay, so here if you have DNA, so you have a +1 region also, which is called as a fidelity. Okay, the lines are okay, no problem. This is a +1 region where transcription start site or a fidelity, and then you have two boxes over here. Okay, one of the boxes here, it is called as a promoter element. Okay, and this proximal promoters and distal promoters are also present over here. Okay, and DNA polymerase in eukaryotes, I told you earlier that the RNA polymerase in eukaryotes, it is of 3 types: the RNAP1, the RNAP2. I told what are the transcripts that would be synthesized by this. Okay, RNA polymerase 1 will synthesize the rRNA 28S, 18S, and 5.8S. Okay, RNA polymerase T2 will be synthesizing the mRNA and snRNA, and 3 will be synthesizing the tRNA and 5S RNA. Okay, these three are differentiated on the basis of sensitivity to, this is base OTBO, on the basis of sensitivity to your alpha amnitin, which is acquired from your mushroom, which is called Amanita muscaria mushroom. Okay, understood. So we have factors involved in your eukaryotic transcription, factors involved in eukaryotic transcription. So what are the factors involved? We have activators, we have basal regulatory factors or transcription factors, and co-regulators. Okay, so you have A, B, and C factors involved. Okay, the first one are called as activators. The second one are called as basal transcription factors, and the third ones are called as co-regulators. Okay, co-regulators, understood. So we have activators. What activators you have? You have SP1, okay, ATF, and CTP. Okay, SP1, ATF, as well as CTP. Understood. And basal transcription factors, we have Tata binding protein, transcription factor 2A, 2B, 2F, 2E, and 2H. These all factors are basal transcription factors. And a co-regulator, we have T transcription factor 2D, which is found from TBP, and 14 TAF. TAF refers to transcription activator factors. Okay, transcription activating factors. Understood. So this all factors you have to remember. Activators, you have SP1, ATF, CTP. Basal transcription factors, you have Tata binding protein, TF2A, B, F, E, and H. Co-regulators, you have TF2D. Okay, understood. So what is the process? First of all, the TBP, the Tata binding protein, this is a Tata binding protein. What it will do, it will recognize the Tata box. Okay, it will recognize the Tata box. Okay, Tata box. Here you have the Tata box here. Okay, Tata box at -25 region here. Okay, and this is the CAAT box at -70 region, and we have GC box also in -110 around. Okay, I'm not writing the numbers, it may vary. Okay, so first of all, let us draw a DNA strand. Okay, this is the +1 region or the transcription start site. Okay, and here you have the Tata box. This is the Tata box or your Hogness box. It is present -25 in case of your eukaryotes, but in prokaryotes you can see that the TAT box or the Tata box is present in -10. Here it is present as -25 region. Okay, then you have the CAAT box at the -10, -70 region, and your GC box. Okay, so what happens that your Tata binding protein is this. Okay, this is your Tata binding protein. Okay, this will recognize the Tata box, and Tata binding protein will help to bind the transcription factor 2D. Okay, this is the transcription factor 2D which is bound to your Tata box. This is a transcription factor 2D. Okay, this will help to bind the transcription factor 2D. Okay, this is the TF2D. Then another enzyme acts, the factor X, which is called TAF. What is TAF? TAF is a transcription activation factor. Okay, what it will do, it will bend the DNA. Okay, it will bend the DNA like this. Okay, it will bend the DNA in this manner. Okay, in this manner. Okay, it will bend the DNA in this manner. So here what you have your Tata binding protein TBP here. Okay, and then your above it you have the TF2D. Okay, TF2D. Understood? Then what happens? Then to it TFA gets attached and your TFB gets attached. Okay, you have TFA and your TFB attached to it. Okay, what is the function of TF2A and TF2B? It will help to stabilize. stabilize the DNA. It will help you stabilize the DNA, bend structure of DNA. Okay, and this TF2B, this binds to a Tata binding protein only. Okay, and this TF2B, this recruits another factor which is your TF2F. Okay, so this TF2B, this will recruit another factor which is your TF2F. Okay, TF2F. Understood? So TF2F is bound to your RNA polymerase. So here TF2F, it is bound to your RNA polymerase here. Understood? This is your RNA polymerase. And the TF2F, after the addition of TF2F, we have the addition of TF2E. Okay, after TF2F, we have TF2E bound to this. Okay, TF2E. This TF2E will recruit TF2H. Okay, TF2H will be recruited here. Okay, then this is the TF2H. What is the action of TF2H? So TF2H has a wide range of functions. H means helicase activity, so it has helicase activity. Okay, it phosphorylates the CTD. Okay, this phosphorylates the CTD, the carboxy terminal domain, okay, of RNA polymerase. Phosphorylates the CTP of RNA polymerase, and it activates the RNA polymerase. Okay, it activates your RNA polymerase. So it is very important for transcription to occur. Okay, it is also helpful in a nucleotide excision repair. Okay, it is also helpful in the nucleotide excision repair, which we saw at that moment. Okay, the disorders and all. Okay, so afterwards, what happens? So what is the sequence of recruitation? We have first Tata binding protein to it. Tata binding protein has TF2D. After TF2D comes, we have TF2A. After TF2A, we have TF2B, and the TF2B recruits the TF2F. This TF2F is binding to RNA polymerase, and TF2E comes. After TF2E, recruits the TF2H, and this TF2H will activate the RNA polymerase. Understood? So we have DABFEH. Okay, this is the sequence of recruiting of the transcription factors. Understood? So afterwards, what happens? This all the complexes, this forms your pre-initiation complex. This forms your pre-initiation complex. Okay, pre-initiation complex. Understood? So after the pre-initiation complex is formed, what happens? All the factors dissociate. All factors dissociate except TF2F. TF2F except TF2F, all factors will dissociate. Okay, so this will look like this something. Okay, this will be the RNA polymerase here. This is the RNA polymerase here, and the factor F, which is tightly bound to the RNA polymerase, present here, and it will start the formation of RNA. Okay, start the formation of RNA. Then elongation will occur. After promoter clearance at that stage. Okay, then elongation will occur. More and more nucleotides will be added. Okay, so 3' to 5' phosphodiester linkages will be formed. Ester linkages will be formed, and addition of nucleotides will start, due to which the RNA will start to form. Okay, then after elongation we'll see the termination. About the termination, so we have, for termination we have two another factors. Okay, for termination of two another factors. So let's say here is our RNA transcript. Okay, so here we have a sequence AAU AACA, AAU AAU AACA. Okay, to this a factor binds. Okay, to this two factor binds in fact. Okay, one is your to this one factor binds, which is called as a CPSF. What is CPSF? Your cleavage and polyadenylation specific factor. Okay, and another factor binds, which is your CSF. This is your cleavage stimulation factor. This both factor combine and recruit another factor, which is your CF. This is your cleavage factor, and cleavage factor is exonuclease activity, which brings about dissociation. Okay, so this is the transcription in your eukaryotes. Understood? So transcription in prokaryotes and transcription in eukaryotes are nearly finished. Okay, this is the transcription process in eukaryotes. Afterwards, let's talk about some post-transcriptional modifications. Okay, let's talk about post-transcriptional modifications. You can get a short note on post-transcriptional modifications also. So what you have 5' capping. Okay, you have 3' polyatailing, okay, or tailing. You can have splicing and RNA editing. Okay, so we will discuss 5' capping, tailing and splicing here. RNA editing will discuss in your gene expression. Okay, so first let's talk about 5' capping. So we have the cap is called as a 7-methyl guanosine triphosphate cap. This is the cap, or it is called as a 7-mg cap. Okay, 7-mg cap, which is attached to the 5' end of the mRNA. So here we have the mRNA here. Okay, and here 7-methyl guanosine cap. Okay, GTP cap. Okay, 5' to 3'. What is the use? That if some exonuclease, it prevents the activity of exonuclease also. It gives stability. It helps in translocation from nucleus to cytoplasm. The RNA has to travel to the cytoplasm. Okay, from nucleus to cytoplasm. Okay, and helps in binding of mRNA, binding to ribosome. So this is the importance. Okay, and the linkage which is adding this, it is the linkage which is forming between the cap and the DNA strand is called as your triphosphate linkage. Okay, then let's talk about, after 5' capping, let's talk about your 3' polyatail. Okay, 3' polyatailing, or this enzyme used in this process is your polyadenylate. This is your polyadenylate polymerase enzyme. Polyadenylate polymerase enzyme. Okay, so we have the signal here. Signal is your AAUAA. Okay, if there is AAUAA signal, okay, then the polyatail formation starts. Okay, so here we have the RNA strand. So to here a tail of AAs are added. Okay, to the 3' end, it is a 5' end. Okay, about 550 to 250 nucleotides of AA are added. Okay, this will prevent the action of your 3' exonuclease. So if there is exonuclease activity, it will prevent the exonuclease activity. It will give the stability, and it will help serve as a site of binding of PAB protein. Okay, it will serve as a site of binding of PAB protein, which we will see in your translation procedure of eukaryotes, which is required for recruitment of 40S ribosomal subunit. Okay, I will tell about it later, so I am not discussing it here. Let's discuss the third, that is your splicing. Okay, so what is splicing? Splicing is basically we have exons and introns. This exon, intron. Okay, so we basically have to remove the exon part and join the remove the intron part and join the exon part. This must be removed. Okay, to join the exons. Okay, E1 and E2, let's name it. Okay, so we can have splicing in two ways. One is intron mediated splicing, and another one is the spliceosome mediated. Okay, spliceosome mediated, intron mediated, and the spliceosome mediated. So intron mediated splicing, it doesn't require any ATP or proteins or factors. Okay, ATP is not required, your proteins, factors are not required. Okay, nothing is required. First what happens is that this is a 5' end, this is a 3' end. Let's say here is one exon E1, okay, and here is another exon E2, and here we have the intron present over here. Okay, so here we have a branch point containing adenine. This is called as a branch point. Okay, then you have the this sequence is called as the 5' splice site. This is called as the 3' splice site. Okay, so this is the branch point, and the 5' and 3' splice sites are there. So what happens is that the branch point, it attacks on the 5' splice site also only. Okay, and the whole part, this whole part gets bent like this. Okay, this get bent like this. Okay, and it detaches from here, and it generates a OH group here. Okay, which is called as a 5' OH group. Okay, it generates an OH group, and after this occurs, okay, this OH group, this produce a OH group. This OH group will again attack on this and will get removed. This whole part, this one will get removed. Okay, this will get removed. Okay, this will get removed. Understood? And this removed part like this, they are shaped like this, and these are called as the lariates. These are called your lariates. Okay, so this is removed, and your exons are joined here. Okay, E1 and E2 are joined here. Understood? So this is called intron mediated splicing. Then another one is your spliceosome mediated splicing. Okay, about the spliceosome mediated splicing, it requires some proteins as well as RNA molecules. It requires 5 snRNAs. Okay, 5 snRNAs, small nuclear RNAs, and 60 proteins. Okay, it is a complex procedure. Okay, this both forms SNRPs, SNRPs, SNRPs. Okay, small nuclear ribonuclear proteins, so 5 snRNAs designated from U1, U2, U3, U4, U5, U4 and U5. Okay, these are the 5. So what happens first is that you have the DNA strand like this, 5' and 3' strand. We have exon 1. Okay, we have the 5' 5' splice site. Okay, and you have the adenine present over here, the branch site, and you have the 3' splice site, and the exon 2 is there. Okay, so what happens is that the U1 get binds to the 5' splice site. U1, so I'm not using the yellow. U1 will get bind to this splice site, and U2 will get binds to the adenine site. Okay, U2 will get bind to the adenine site. Okay, then after inactive complex of U4, U5, U6 are removed. Okay, then after U1 and U4, these are removed. U1 and U4 are removed. Due to this, there will be loop formation will be there. As U1 is removed, the same way it will form the loop, the lariat. Okay, the same way it will form the lariat, and due to this loop formation, the lariat will be removed. Okay, the lariat will be removed, and there will be joining of two exons, E1 and E2. Understood? So these are the post transcriptional modifications. Let's talk about your translation procedure. Okay, translation. So we are first talking about translation in your prokaryotes. Okay, translation in your prokaryotes. So talking about translation in prokaryotes, so we have requirements in prokaryotes. We have to get mRNA, we have to get tRNA. Okay, we want ribosomes, and the prokaryotic ribosomes are 70S, and 70S is 50S and 30S, and 50S has 23S and 5S, and 30S has your 16S. Okay, this has about your 34 proteins, and this is about your 21 proteins. Understood? What are the factors used? Factors for initiation, for elongation, and for termination. What are the factors used? Okay, so for initiation factor 1, 2, 3, 4, 5 and 6 are used. For elongation, we have EFTU and EFG. Okay, and for termination, we have release factors 1, 2 and 3. Okay, these are the factors that will be used in your prokaryotic translation. Okay, so first First there will be charging of tRNA. Okay, so what happens in the charging of tRNA is that there will be first amino acid activation. Okay, so the amino acid is not activated, combined ATP in the presence of amino acyl tRNA synthetase. This will form ENZ AMP AA complex. Okay, this is your activated amino acid. Okay, this activated amino acid will combine with your tRNA. Okay, and form your tRNA. tRNA has your CCA arm which accepts the amino acid, so we write CCA, and it attaches to the OH of amino acid, so amino acid OHAA amino acid. Okay, this whole part is your charged tRNA. Okay, so charging of tRNA is done. Then the second step in this process is initiation. So what happens in the initiation? First what happens, I am writing it A. First is the ribosomal dissociation. In this the 70S in the presence of initiation factor 6, very important, initiation factor 6, it splits into your 50S and into your 30S. Understood? Your 50S and your 30S. Okay, then afterwards this 30S, this combines with the initiation factor 1 plus initiation factor 3. Okay, initiation factor 1 and initiation factor 3 to form this 30S initiation complex. Okay, so B 30S initiation complex formation. Okay, so 30S initiation complex, how does it look like? So just imagine this as the mRNA. Okay, and this is your 30S subunit attached to here. Here you have 1 and here you have 3 attached to it. Okay, this is 30S initiation complex formation. Okay, then C1 is a formation of ternary complex. So what is ternary complex? Let me tell you. So ternary complex is formed by the tRNA which will contain the 5' methionine, the formyl methionine. Okay, formyl methionine. So this is called as formyl methionine tRNA, formyl methionine tRNA. Okay, it will combine with IF2 and a molecule of GTP, to form the ternary complex. Okay, only IF2 and the molecule of GTP to form the ternary complex. Okay, so 30S initiation complex formation will occur later, so you can't write here. Okay, basically the ternary complex is formed. This is a ternary complex, and afterwards you just name it B. Okay, and then you just name it C. So in C point, it's the formation of the 30S initiation complex. So what happens is that here is the 30S subunit. Okay, here you will attach your 5' methionine or your formyl methionine tRNA. Okay, and to it factor 1 and factor 3 are already attached here in this step, IF1, IF3. Okay, and your IF2 in your ternary complex will be attached. It will be attached to the GTP molecule here. Okay, understood? This is the 30S initiation complex formation. Afterwards what happens, the 70S ribosomal subunit attaches to here. Okay, sorry, the 50S ribosomal subunit attaches to here, not the 70S. Okay, so 50S, here's the 5', all the factors will dissociate. Okay, this 1, 2 and 3, this will be hydrolyzed by the GTP, and this structure will be formed. This structure is called as your option and D, that is your 70S initiation complex. Okay, 70S initiation complex will be formed. After the 70S, it has 3 sites, you know. Okay, E site, P site and A site. So this is the exit site, this is the P site or peptidyl transferase site, and this is the A site or acceptor site. Okay, so then what will occur? Then after the initiation step, okay, the initiation step is finished over here. Okay, then after we have the termination step. Sorry, not the termination, I'm saying termination, why I don't know. Elongation step, elongation factors, there will be 2 elongation factors, EFTU, okay, and EFG. What will EFG do? EFG will provide energy for translocation. This will provide energy for translocation Okay, translocation. Okay, so first what happens is that to this A site, P site and E site, okay, E site, P site and E site, first this will come, okay, this will come and attach to this A site only, okay, the ternary complex. In this ternary complex it is attached to your A site only, okay, to the A site. Then after second amino acid will come, and second amino acid will carry another amino acid. This will shift the mRNA will translocate. This translocation will occur, and this will go here, okay, and this will come here. After another translocation, this will go here, okay, and give the amino acid here to this peptide bond formation will be there in peptidyl transferase site, the P site, okay. Then another amino acid will come, this will go here, and again on shifting it will form another amino acid. So there is the formation of amino acid chain by chain in your P site, okay, and the energy is provided for translocation. The energy is by EFG, okay. Then there is termination, okay, after elongation there is termination, okay. If you encounter, if the tRNA it encounters a stop codon, so what are stop codons? Stop codons are UAA, UGA and UAG. These are stop codons. When it encounters a stop codon, then termination will occur, okay, like AUG here it was, okay. Then you have that factor here, the amino acids are synthesized 1 2 3 4 5 and all, okay. This protein is getting synthesized. So here we can have a release factor which is an analog of a tRNA only, okay. This is a release factor of prokaryotes, which is an analog of tRNA, okay, and this release factor this will cause the hydrolysis on occurring of the stop codon UAA, like it's a stop codon. It will cause the hydrolysis here, and the chain will be terminated, the protein will be removed out, and the proteins are folded by some special proteins like chaperones, okay. Chaperones are folding proteins which will fold the protein. Understood? So we have the inhibitors of protein biosynthesis. In your prokaryotes, okay. So the prokaryotic inhibitors. So for prokaryotic inhibitors, in prokaryotic inhibitors of protein synthesis, we have tetracycline. We have your chloramphenicol. We have erythromycin and your aminoglycosides. Okay, so tetracycline example is your doxycycline. Okay, doxycycline is a tetracycline, and this tetracycline binds to a 30S subunit and prevents the attachment of amino acid tRNA to the A site, okay. It prevents the attachment of tRNA to A site, okay. Prevents attachment to A site, okay. Tetracycline is prescribed as a broad spectrum antibiotic. It is a broad spectrum antibiotic, okay. And we have chloramphenicol for it is an antibiotic for your typhoid, okay, or for your enteric fever, or for also used for your meningitis. It binds to a 50S subunit, and it inhibits the activity of peptidyl transferase. So basically it is blocking the peptide bond formation. Erythromycin is used as a upper respiratory tract infection or lower respiratory tract infection. It is used in this, okay. It also binds to your 50S and prevents attachment to your P site. tRNA to the P site. Aminoglycosides, for example, you have streptomycin, you have gentamicin, okay, you have neomycin, you have amikacin, okay. These all are aminoglycosides. So these are gram bacterial antibiotics. Okay. Streptomycin basically used in the TB treatment, tuberculosis treatment. Okay, this also bind to the 30th subunit, and this inhibits the initiation complex formation. Okay, this misread the mRNA. Okay, and also prevents translocation. There's a wide range of functions are there of your aminoglycosides. Okay, so these four are your prokaryotic inhibitors of your transcription. Okay, these four are the prokaryotic inhibitors of your transcription procedure. Okay, so let's now move to your translation in your eukaryotes. Okay, eukaryotic translation. So let's talk about your eukaryotic translation. Eukaryotic translation. So in eukaryotic translation, first we will have activation of tRNA. Okay, so let's activate the tRNA. How it will activate? A is bind with ATP in the presence of amino acyl tRNA synthetase. Okay, it will form your ENZAMP AA. It is an active amino acid. This will combine with your tRNA and will form your tRNA CCA OH AA. Okay, this is your activated tRNA. Okay, then there will be initiation of translation. Okay, so first there will be mRNA activation. Okay, we have to activate the mRNA first. So in mRNA activation, here's the mRNA 5' and 3'. Okay, here you have the AUG sequence. Okay, so there will be a factor called as a 4F. Okay, 4F will combine 4G plus 4A plus 4E factors. Okay, this 4A will remove the secondary structure of RNA. Okay, RNA is present secondary structure, then it will remove the secondary structure of RNA, and E will help in recognition of cap. Okay, it will help in the cap recognition. Okay, so 4F factor will bind over here. Okay, let's draw it again. So this is the 5' and this is the 3' end. 4F factor will bind to the 5' end 4F factor. Okay, on binding of the 4F factor, we will see the binding of 4B and plus PAB. What is PAB? PAB is the poly A binding protein. So PAB will bind to here. Okay, and the 4B will bind to the AUG region. This is the AUG. Okay, this will bind to the AUG region. So it will look like something this 5'. This is a 3'. This is a 4F binding over here. The 4F factor binding over here, and this is the AUG region over which the PAB is binding. Sorry, the 4B will bind. Okay, 4B will bind, and the PAB will bind to the poly A tail. This will bind your poly A binding protein. Okay, poly A binding protein we saw in your post transcriptional modifications. Okay, and this 4B will start synthesizing a 18S rRNA transcript. Okay, this will start synthesizing a 18S rRNA transcript, and this region, to be specific, this region, okay, this coding region is called as a Kozak consensus sequence. In prokaryotes, it is the analogous to your Sine-Delgarno sequence. Okay, understood. So 4B, what is the function of 4B? It recognizes the AUG sequence. And what is the function of PAB? PAB will recruit the 40th subunit. Okay, it will recruit the 40th subunit to the mRNA. Okay, then after mRNA activation, then there will be the step which is called as a ribosomal dissociation, in which the 80S ribosome will split into 40S and your 60S. Okay, in the presence of eukaryotic initiation factor 6. Okay, there was prokaryotic in that initiation factor 6. Okay, here in prokaryotes EF6 we wrote. Okay, the IF6 is we wrote. Okay, initiation factor 6. Here I have eukaryotic initiation factor 6 only. Okay, then afterwards what will happen? Then there will be a formation of ternary complex. Ternary complex here will also contain your methionine tRNA, MET, not formal methionine, only methionine tRNA. EF, eukaryotic initiation factor 2, that's your GTP, same thing. Okay, this is a ternary complex. After the ternary complex, there will be a formation of 43S pre-initiation complex. Okay, formation of 43S pre-initiation complex, in which there will be 40S ribosomal subunit plus eukaryotic initiation factor 1 and 3. Okay, 1 and 3 plus your ternary complex. Understood? So they will combine. Okay, so I was telling you that 43 pre-initiation complex is found where 40S ribosomal subunit, your eukaryotic initiation factors 1, 3, okay, and your ternary complex. Okay, so this is your 43 initiation complex, and methionine is there, 1 must be there, 3 is there, the 2 is there with GTP attached to it. Okay, then there will be formation of your 43S initiation complex. This was a pre-initiation complex. Then there will be initiation complex formation. Just you take this and add your mRNA. Okay, mRNA. You will see the formation of a 43S initiation complex. 1, 2, 3 GTP attached to it, methionine to it, and this mRNA 5' to 3'. Okay, understood? This is the 43S initiation complex. Again, the large ribosomal subunit attaches to it. Okay, so the large ribosomal subunit, the 60S subunit, that is, okay, plus your 40, okay, this is 48S, sorry, I'm sorry, 48S initiation complex, which is formed by this. Okay, and the large subunit for plus 43S initiation complex will give rise to 80S initiation complex. Okay, so this is the for 60S which is attached, this is the 40S which was already there, 5' and 3'. All other factors will dissociate. Okay, there will be the methionine RNA, tRNA. Okay, and on the factor 5 will be there. Okay, here the factor influencing will be eukaryotic initiation factor 5, EIF 5 will be there. Okay, only 5 will remain, all others will dissociate. Okay, and this complex formed will be called as active 80S initiation complex. Okay, so initiation is completed here only. Ribosomal dissociation, formation of ternary complex will be there. 43 as pre-initiation, 48 as initiation, 80 as initiation complex. Okay, formation, and all other factors will dissociate. Then we come to elongation. So in elongation, first there will be tRNA attachment, tRNA attached to the A site, and there will be peptide bond formation. In the same way as we saw in your prokaryotic elongation. Okay, then translocation will occur. Okay, so this energy for peptide bond formation is given by EEF to eukaryotic elongation factor 2. This will provide energy to the peptide bond formation, and tRNA attachment is facilitated by eukaryotic elongation factor 1. Okay, this will provide energy for tRNA attachment to the A site. Okay, this just a clinical, this eukaryotic initiation elongation factor 2, this is inactivated by a toxin called as a diphtheria toxin. So diphtheria toxin inhibits your eukaryotic elongation factor 2. Understood? Then go to the termination part. So termination is just by release factor 1 and 3. Okay, so let's say this is the stop codon UAA. Okay, so here release factor 1 will act. Okay, this is the RF release factor 1. Okay, and to it binds release factor 3, and this whole complex will hydrolyze the peptide bond. This will hydrolyze the peptide bond. Understood? Next we'll talk about the inhibitors of protein synthesis. Inhibitors of protein synthesis in eukaryotes. Okay, so So first of all, there will be diphtheria toxin. What I am dating, diphtheria toxin. This diphtheria toxin is secreted by the Corynebacterium diphtheri, okay, which will cause the nasopharyngeal infection. Okay, this causes a nasopharyngeal infection. This will inhibit the eukaryotic elongation factor 2, and this will cause by the method which is called as a ADP ribosylation. Okay, ADP ribosylation. By ADP ribosylation, it inactivates the eukaryotic elongation factor 2, okay, which is required for translocation. Then we'll another inhibitor is there, that is a ricin, RICIN, ricin. This binds to a 60S ribosomal subunit. This binds to a 60S subunit. Okay, it is a protein from castor beans, from castor. Okay, from castor beans. It is binding to 60S and removes the adenine nucleotide from the 28S rRNA. Okay, it removes adenine nucleotide, removes adenine. Okay, it removes the adenine from the nucleotide. Okay, blocking the translation process, not the translocation, translation process. Okay, then we have cycloheximide. Cycloheximide. So cycloheximide also binds to a 60S subunit. Okay, and it inhibits the peptidyl transferase activity. It inhibits the peptidyl transferase activity. Okay, just see that in prokaryotes also, the chloramphenicol, okay, this was also inhibiting the activity of peptidyl transferase, and in eukaryotes your cycloheximide, this inhibits your peptidyl transferase activity and blocks your translation procedure. And there is an inhibitor called as a puromycin. This puromycin will it's a structural analog of your tyrosinyl tRNA. Okay, it causes premature termination of protein synthesis. It terminates the protein synthesis in, this is important, in eukaryotes as well as prokaryotes. Okay, in both. Okay, this is the inhibitor protein synthesis in both. Understood? So this completes your translation in your eukaryotes. Let's talk about some post-translational modifications. Talking about post-translation modifications, we have two types. Okay, one is called as a proteolysis, and another is called as a covalent modifications. Okay, about the proteolysis, it has about three types, called as first one is the removal of initiator methionine. Okay, to remove the initiator methionine, we do proteolysis, and this is a post-translation modification. It involves activation of proteins, activation of inactive forms of proteins called as a proteins. Okay, proteins like your trypsinogen is converted into trypsin, chymotrypsin and chymotrypsin, procarboxypeptidase into carboxypeptidase, proelastase into elastase, pepsinogen into pepsin, many of them. Okay, then cleavage of signal peptide. So you know, signal peptide is a molecule that is attached, which is important for the protein targeting, and sorting mechanisms. Okay, this is also cleaved by proteolysis. Understood? So after this, we can have covalent modification. First is the addition of carbohydrates, which adds to the antigenicity, of the protein. Okay, this occurs in your Golgi apparatus, where the three sides of Golgi have different functions, like the cis side, the medial side, and the trans side. Okay, the cis side of Golgi is helpful in removing the mannose. The medial side in fucose and N-acetyl glucosamine addition, fucose and N-acetyl glucosamine addition, and the trans side in galactose and the sialic acid addition. Okay, And this addition of carbohydrate also is responsible for the cell to cell recognition. Okay, cell to cell recognition. Understood? Then another thing is your prosthetic group addition. Okay, addition of carbohydrate we did. Then addition of prosthetic group like your biotin, your heme. Okay, prosthetic group addition. Then third thing is your phosphorylation and dephosphorylation. Okay, so you need to understand which enzymes are phosphorylation, which activate phosphorylation, which are inactivate phosphorylation. Okay, so for if I give an example of glycogen phosphorylase is activate at phosphorylation phase. Okay, and your glycogen synthase is active in the dephosphorylated phase. Okay, then we have another modification, that is your SS group addition. Example, the disulfide bond formation. Example is your insulin. In insulin maturation, the SS bonds are formed. Okay, then for example, another example may be OH group addition, which is called as your hydroxylation. So formation of proline, OH group is added, so you form hydroxyproline. Okay, and lysine, you form hydroxylysine. Okay, then you have sixth, gamma carboxylation. Okay, with in your formation of your vitamin K and your osteocalcin. Okay, then you have acetylation, and again you have isoprenylation. Isoprenylation is important in the cholesterol synthesis. Okay, understood? So these are the post translational modifications in your eukaryotes. Okay, then we will talk about the gene expression, control of gene expression. This is an important topic, control of gene expression. Okay, in this control of gene expression, we'll talk about gene regulation. If we talk about gene regulation, we can see it has a positive regulation, negative regulation. Positive regulators are called as your enhancers. Okay, enhancers are positive regulators, and your repressors or regulators or your silencers are your negative regulators. Okay, we have two types of genes too, also called as your housekeeping genes. Okay, these are expressed as the constant rate and maintain the basic cellular functions, while we have regulated gene or regulatory gene. Okay, which express only in certain conditions. Okay, what is the site of gene regulation? So in prokaryotes, the gene regulation is at transcription level. So we will discuss the gene regulation at transcription level only, but in eukaryotes the gene regulation is at transcription level, at post transcriptional level, at post translational level also. Okay, the transcription level is the most common. Okay, we have some cis elements and trans acting factors. So cis acting elements, these cis acting substances or elements, these are present on DNA, and these are regulatory DNA sequence. So this is a cis acting element. Okay, and to this some regulatory factors bind. Okay, regulating factors, the trans acting factors, this bind to the cis acting factors. Okay, these are also regulatory in nature. Okay, so binding of proteins to DNA, binding of proteins, any protein, if it is to be bind to the DNA, okay, it is done by structural motifs. Okay, we have many motifs like a zinc finger motif, okay, leucine zipper motif, or your helix turn helix motif. So you can refer the Lippincott textbook for more details on this. Okay, these are not your core syllabus part. Zinc finger motif example, binding of your transcription factor 3A. Okay, binding of your transcription factor 3A, okay, is via zinc finger motif. Your lysine zipper motif example is your phos and June proteins, okay, and helix turn helix motif like your crow protein, which is seen as a regulatory role in your molecular switching mechanisms. Okay, and the molecular switching mechanism, which is beyond the scope, but I will just tell a little. Okay, the main thing in the regulation, the control of gene expression, is the lac operon, which is asked frequently. This is a common concept, but it is frequently asked. So let us discuss the lactose operon or the lac operon. Okay, so lac operon, the inducer is a lactose. Okay, the inducer is the lactose. The real inducer is the allolactose. So it is a linear array of genes involved in the metabolic pathway described by your Jacob and monad. Okay, so we have three structural genes here. Okay, like lac Z gene, the lac Y gene, and the lac A gene. The lac Z gene codes for beta galactosidase, okay, which converts the lactose into galactose or glucose. Okay, the lac Y gene, which codes for permease, increase the permeability of cell for lactose to enter the cell. Okay, then lac A, we have thiogalactoside transacetylase, okay, which acetylates the lactose, Understood? The function of acetylation of the lactose is yet unknown, as written in your Lippincott textbook. Okay, so let's draw the linear array of genes present over here. Okay, if I to draw the genes. Okay, so this is your lac I gene, which is called as a repressor gene. This is a cap site. Okay, this is a promoter. Let's draw the promoter region here. This is the promoter region. Okay, then there is an operator region here. Let's draw the operator region. This is the O region. Okay, then the Z, your Y, lac Z, your lac Y, and your lac A are present. Understood? So let's you know what is cistron? Cistron is the smallest unit of gene expression. This is the smallest unit of gene expression. Okay, cap means catabolic activator. Cap means catabolic activator protein. Okay, catabolic activator protein, otherwise called as a CRP. This full form is camp regulatory catabolic activator protein or camp regulatory protein. This is the full form of your cap region. Okay, so we'll see what the role of cap is. Okay, so let's take the cases that when glucose is present, if your glucose is present and lactose is absent. So what will happen if the glucose is present? The bacteria is happy. Okay, the bacteria is happy because glucose is present. Okay, it will be able to take up the glucose, so it doesn't need the lac operon. Okay, so bacteria will use the glucose only. So operon will be repressed. How the operon is repressed? Let's see it. Repressed or switched off. This is called as a negative regulation. So if glucose is present, there will be negative regulation of the operon. Okay, so how it is done? Let's see. So if here is the thing. Okay, the lac I gene is there. Let me copy and paste the linear array. I can't draw the linear array many a times, such like I have to draw at least 2 times. So why I should draw it? Let me copy paste. Okay, so negative regulation I am showing you. So in this case, what will happen is that the lac I is a constitutive gene. This is a constitutive gene. Just note that constitutive gene. That means it is expressing at a constant rate. Okay, so it will synthesize your RNA. It will synthesize your RNA. RNA will synthesize your repressor protein. RNA will synthesize your repressor protein. Repressor protein. Okay, this repressor protein will be activated in the absence of glucose, because it in the absence of lactose, because lactose is absent, so it gets activated. Okay, like this, a tetramer is produced. Tetramer. Okay, this tetramer will go and bind to the operator region. This tetramer will go and bind to the operator region. Okay, and thus the RNA polymerase can't bind. The RNA polymerase, this is the RNA polymerase, can't bind the promoter region and continue the transcription. Okay, the translation, sorry. Understood? So due transcription is there, yeah, transcription, sorry, so I got confused a bit. Okay, no problem. Lag Z will not express, this will not express, this will not express. Why? Because the this part is blocked by the repressor protein molecules. Okay, due to which the RNA polymerase can't bind, and the transcription transcription is just repressed. The operon is repressed. Okay, understood? Then what is the role of cap here? That this is the cap protein. This is the cap protein. Okay, catabolic activator protein, catabolite activator protein, cap protein. Okay, glucose is present. As glucose is present, adenyl cyclase is inactive here. Adenyl cyclase is inactive here. Okay, so cap protein is present in the unbound form. Okay, so cap is not present here, so cap will not bind to the cap site. Okay, this will be there in the unwound form. Okay, this inactivation by adenyl cyclase, due to which the cap protein is unbound, this is called as a catabolite repression. Okay, term is called catabolite repression. Understood? What are the phenomena that are happening? Understood? So this is the first case. Let's say if you have lactose and no glucose. If you have lactose and no glucose, lactose present, okay, and glucose is absent. So here the lac operon will be induced. Lac operon will be induced. Okay, this will be on. This is called as your positive. This will call as your positive regulation. In this positive regulation, what will happen, that if here is your linear array, let's draw the linear array again here. Okay, so what happens here is that, of course, the lac-I is a constitutive gene. It will produce the mRNA. Okay, mRNA will obviously produce the receptor protein. Okay, and the receptor protein, in the presence of lactose, the lactose present. Okay, let's say these are the molecules of lactose. Okay, this will bind to the repressor protein. Okay, this will bind to the repressor protein and render them ineffective. This now can't bind to the O. The tetramer is not produced. It can bind to the O. So easily the RNA polymerase, here the RNA polymerase is there. This can easily attach to the P site. Okay, and continue the transcription process. Due to the transcription, lag Z will give beta galactosidase. This will give the mRNA. This will give the mRNA for permease. This will give the mRNA for transacetylase. So this mRNA, composing of the combination of the products of the three genes. Okay, this is called as a polycistronic mRNA. Okay, and this will give your beta galactosidase, your permease, okay, and your transacetylase, thiokinase transacetylase. Understood? I was seeing something. Did I tell you what are the things synthesized by them? Okay, thiogalactoside, transacetylase I told, okay, permease I told, and beta galactoside, yes, I have told. Okay, no problem. They are synthesized. Understood? Another case is there. Another case is, let's name it 3, that lactose is also present and glucose is also present. So what will happen if lactose and glucose are both available? Then bacteria will prefer the glucose. Okay, bacteria will take this. Bacteria prefers glucose. Bacteria prefers the glucose. If the bacteria prefers the glucose, then the same case, then there will be the same case as if glucose is present. Okay, same case as 1. Understood? So lactose operon is done here. Okay. Yeah, I have to teach another thing, that cap. What is the effect of cap if lactose is present and glucose is absent? Glucose is absent, your adenyl cyclase is active, adenyl cyclase is active. So what will it do? That this is the your cap, catabolite activator protein. Okay, and it will bind with the CAMP, it will bind with the CAMP. Okay, so here we have the CAP and you have the CAMP. Okay, and this will bind to the cap region. Okay, this will bind to the cap region. Understood? In the absence of glucose. So this is called as positive regulation. After this we have another operon to discuss, that is your tryptophan operon. So talking about tryptophan operon has five structural genes. Understood? Okay, so it has five structural genes. Okay, the TRP operon has five structural genes. So it has also a promoter region. This is the promoter region of the TRP operon, and structural genes are labeled from A to E. Okay, so this is TRPE, okay, TRPD, TRPC, okay, TRPB, and the TRPA. Okay, this is these are the five structural genes present over here. Okay, after this presence of genes, okay, what happens is that it is subject to the negative control. First, let's take the case that TRP is plenty, tryptophan is plenty. Okay, if you have plenty of tryptophan, there is no need of synthesis. Okay, so the operon is switched off. Okay, by an attenuator. The main mode of synthesis, the tryptophan operon works, the main mechanism is called as attenuation. Attenuation. So what is attenuation? Attenuation is the formation of a hairpin structure in the RNA. Attenuation is formation of a hairpin structure in RNA. Okay, if it is plenty, then the hairpin structure will form in the RNA. Okay, if it is synthesized, the hairpin structure will form in the RNA, and this hairpin structure will prevent the transcription process. Okay, it will prevent the formation of tryptophan. Okay, in the tryptophan mRNA. Okay, the tryptophan mRNA, from this and this is the DNA strand, you know. Okay, so there must be the mRNA TRPE, okay, TRPD, then the TRPC, the TRPB, and the TRPA. Okay, so you will see that in the mRNA, there will be a hairpin loop-like structure formed. Okay, hairpin loop-like structure formed, which will prevent the tryptophan to be synthesized. Okay, then what will happen if case 1 is done? Case 1 is when tryptophan is plenty. Okay, where I wrote that tryptophan is plenty. Yeah, case 1. Let's discuss the case 2. Tryptophan is scarce. Okay, we need tryptophan. The tryptophan is scarce, the operon is switched on. Lack of tryptophan is that the ribosomes will stall at 2 tryptophan codons at 5' end. Okay, the ribosomes, there will be presence of ribosomes at 5' end. The ribosomes will be stalled at here. Okay, the ribosome will be stalled at here. Okay, at the 2 tryptophan codons present at 5' end. Due to this stalling, it will cover attenuation site. Okay, this will prevent the attenuation. It will cover the attenuation site, so attenuation won't occur, and the tryptophan synthesis will continue. Go on continuing. Okay, so this is called as your tryptophan operon. Okay, Then we have a genetic switch of bacteriophage lambda also. So bacteriophage lambda, this has its lytic phase and a lysogenic phase. Okay, just I'm telling it in very that very less details. Okay, this is called as the molecular or genetic switching in bacteriophage lambda. Lytic phase and lysogenic phase. In lytic phase, the bacteria is very virulent. Okay, in lysogenic phase it is dormant. Okay, so in unfavorable conditions, when nutrients and sunlight are absent, okay, these are absent, nutrient and sunlight are absent, then what happens is that the OR1 gene, okay, OR1 repressor protein will bind to this. Okay, the repressor protein will bind to OR1 gene, and the OR1 gene will suppress the gene for CRO protein. There will be no CRO protein, and the CRO protein is required for the synthesis of the viral machinery. Okay, so due to no CRO protein, okay, the bacteria will lie dormant. Okay, bacteria will lie dormant. Okay, and dormant, and lysogenic phase will occur. Okay, if the nutrients and sunlight will be present, if these both are available, okay, then the CRO protein is synthesized, and the CRO protein is not repressed mainly. Okay, and the CRO protein synthesis, and bacteria continues its replication. Bacteria continues replication, which is called as a lytic phase. Understood? So this is the control of gene expression in prokaryotes. Let's discuss the control of gene expression in eukaryotes. Control of gene expression in eukaryotes also. So in eukaryotes, it are mainly regulated in many levels. Okay, first let's discuss it at DNA level. Okay, let me talk about the levels. So this is a DNA, then hnRNA formation. The process is called as transcription. Okay, first is at DNA level it is regulated, at transcription level it is regulated, then hnRNA to here post transcriptional modifications occur. So it at this level it is regulated. mRNA is formed due to this. Okay, then translation will occur. Okay, and protein will be formed. At this level it will be regulated, and in protein the post translational modifications will occur. Okay, at this level it will be regulated also. Okay, so there are 5 levels of regulation of gene expression as per your eukaryotes are concerned. Okay, at DNA level, what are the modifications? Okay, at DNA level, so first one is epigenetic modification. Epigenetic modifications. So epigenetic modification we have 2. First is the acetylation of lysine H tails. Okay, due to acetylation, it will decrease the positive charge. It will decrease the interaction between histones and DNA. Okay, and decrease histone DNA interaction, and this is increase the gene expression. Okay, and second one is the methylation of histone. Okay, so we have methylation of histone proteins, okay, which will decrease your gene expression. These are the epigenetic modifications. Then another thing is called as a gene amplification. What is gene amplification? Is that the gene is amplified, like for example a gene for DHFR. Okay, gene for DHFR is there. Okay, the A gene and B gene here. Okay, if this is amplified, will produce something like this. Okay, here the gene is getting amplified. This is called gene amplification. Then let's talk about another modification which occurs at the DNA level only. So epigenetic modification, you have Gene amplification, another one is called as a gene rearrangement. It's called as a gene rearrangement. Gene rearrangement, what is gene rearrangement? So you can just get an idea that this is a immunoglobulin IgG. This immunity is the main part here. Okay, this is your variable region, constant region, variable region, constant region. So you have around 300 B1 from B1, B2, like this you have around 300 genes. Okay, and from B1 to Bn, okay, you have around 300 B genes. Okay, then around for J region, okay, for J region, J1, J2, J3 up to J5, you have up to J5 5 J genes. Okay, for V genes you have 300, J genes you have 5 in number, and D genes also, D1, D2, D3 up to 10. Okay, D1, D2, D3, these are up to D10. Okay, so from this various combinations can be generated, like V2, J3, D3. This one is a combo, like V20, D1 and J2. This is one combo. Okay, so many combinations can be generated. This is called as a so this will produce different light chains for IgG. Okay, this is one thing. So the changes at the DNA level are done, the regulation at the DNA level. Then regulation at a transcription level. Regulation at transcription level is mainly by the regulatory elements like promoter and promoters and enhancers. Okay, promoters and enhancers, these are some promoter elements. Like promoters for regulatory elements, you have enhancers and other elements like your transcription factors and DNA binding motifs, which I taught, the leucine zipper motif, the DNA the helix turn helix motif, and another loop motif is that the helix loop helix motif, and a leucine zipper motif. Okay, and transcription factors, transcription factors, you have many transcription. The TBP is there, then comes TF2D. Okay, TF2D recruits TF2A and TF2B, which stabilize the DNA. TF2B, okay, they recruit the TF2F, which is tightly binding to your DNA polymerase, and TF2F recruits TF2E, which recruits TF2H, which is helpful in nucleoside excretion repair, helicase activity, and also TF2H is useful, okay, in that manners. Okay, then we'll discuss about regulation at post-transcriptional modification level, okay. At this level, what are the modifications? These are the transcription level. We have promoter enhancers, transcription factors, and DNA binding motifs. At post-transcriptional level, okay, at the post-transcriptional level, we have the modifications, which is called as a splicing. The splicing. Okay, so we can have selective splicing or alternate splicing also. Selective splicing or alternate splicing. Okay, earlier we saw about any other type of splicing, like intrinsic splicing and spliceoso-mediated splicing. Okay, here in selective splicing, the exons are just excised selectively. Okay, these exons are excised selectively and they are formed like this. Okay, E1, E2, E3, E1 and E2. Okay, but in alternate splicing, it can have 5' or 3' ends, like each exon may have two parts, 3' part or 5' part. So it may be cleaved in any other combinations also, which will produce different mRNAs. Okay, then after splicing, another modification is your mRNA editing, which we will discuss in a while. Okay, mRNA editing and your mRNA stability. So let's talk about these two terms. Okay, this newly encountered terms, mRNA editing and about mRNA stability. So splicing, mRNA editing, and mRNA stability are three modifications at the post-transcriptional level. Okay, at the post-transcriptional level. So let's talk about this. Let's talk about the mRNA editing. Okay, so very famous example for mRNA editing is a ApoB48 and ApoB100. Here the chain growth is stopped at 48%. Okay, 48%. Protein synthesis is stopped at 48%, but here the protein synthesis has occurred till 100%. Differences are ApoB48 are produced in intestine, okay, intestine, and ApoB100 is produced in liver. Okay, actually what happens in the place of CAA, UAA is encountered at your 666th position. Okay, due to UAA, as it is a stop codon, so it will stop the protein synthesis at 48%. Okay, but in ApoB100 there is no stop codon, it is produced by CAA only, and it goes on synthesis, so 100% is synthesized. Okay, this is called mRNA editing. Talking about mRNA stability is yet another phenomena, mRNA stability. So talking about mRNA stability means that in for example in iron deficiency anemia, example is your iron deficiency anemia. In your iron deficiency anemia, what happens is that this is a 5' end and this is a 3' end. So in iron deficiency anemia, okay, if iron is decreased, okay, so if iron is decreased, in mRNA some loops are generated like this. These are called as IRE. These are called as iron responsive elements. This IRE will bind if iron is decreased. If iron is decreased in the body, okay, then it will produce IRP. It will produce IRP. IRP increase the stability of mRNA. Okay, this bind to the IRE. Okay, IRP bind to the IRE and increase the stability of mRNA, which will lead to transferrin receptor synthesis. Increase, and more iron will be reabsorbed. Okay, will be stored so as to compensate the iron deficiency in the body. Okay. Then we have the regulation at translation level. Okay, regulation at translation level is translation level we saw here. Okay, regulation at this level, translation level, is via mainly your initiation factor 2 and your eukaryotic initiation factor 4E. Okay, 4E is the cap recognition part, Okay, EAIF2 is important for your initiation process. Okay, eukaryotic initiation factor 2 is required for your initiation factor. Okay, so this completes your regulation of gene expression in eukaryotes. Okay, now let's go to the recombinant DNA technology or the genetic engineering. Okay, rDNA technology and genetic engineering. Okay, so talking about this genetic engineering or gene cloning or gene manipulation, what is the step? So first you take a cell, okay, you treat it with detergent, okay, treat it with cell detergents, okay, not the detergents you use at home, okay, then with phenols, with proteases and ribonucleases. Okay, so as to remove the proteins and ribonucleic acids, okay, RNAs. Why is this important? Because we need DNA, not the RNA or proteins. Okay, then you do it ultrafugation is done. Then it is precipitated by using chilled ethanol, and the required DNA is extracted. Okay, this DNA is treated with the restriction endonucleases. Okay, and they are separated by electrophoresis. This is again identified by and visualized by Southern blotting. You know the snowdrop phenomenon, southern, northern, and snowdrop. Okay, SNOWDROP. So southern blotting is for DNA, northern blotting is for RNA, and your western blotting is for your proteins. Okay, so this is snowdrop phenomenon identified by the southern blotting, and the desired DNA has sticky ends now, Okay, has sticky ends. Okay, so we have to generate the DNA with sticky ends. Now let's take the prokaryotic vector which has desired characteristics here, desired characters. Okay, such a desired character. Okay, so prokaryotic vector is desired characteristics. Okay, and the prokaryotic vector is treated with EDTA as well as lysozyme so as to break its cell wall, and due to this the cell wall is broken. Then you treat it with sodium lauryl sarcosynate, sodium lauryl sarcosynate. Then ultrafugation is done, electrophoresis is done, electrophoresis is done, and the plasmid is acquired. Okay, you acquire the plasmid. Then you have to cut the plasmid using the same restriction endonuclease as you cut the DNA. Okay, so as to generate complementary sticky ends. Okay, so you cut the plasmid like this with sticky ends. Okay, then you take this desired piece of DNA, okay, this DNA having the sticky ends and this plasmid, and attach it here. Okay, you attach it here. Okay, this is called as a recombinant. This is called as your recombinant or hybrid or your chimeric DNA. Okay, and this you can transfer the recombinant or the chimeric DNA into the host cell, by gene transfer methods. We'll see how these gene transfer methods occur: liposome mediated gene transfer and the gene gun. Okay, the ballistic methods and all. Okay, and this your chimeric DNA is again incorporated here, and the host cell, when it expresses, it will give you your desired protein. Understood? So these are the steps of your recombinant DNA technology, which I told you in short. Okay, then what are the enzymes needed? So what are the enzymes needed in recombinant DNA technology? We need nucleases. The exonucleases or your endonucleases. Okay, endonucleases. So we require the restriction endonucleases, which are called as the molecular scissors. Okay, the 3 types, 1, 2 and 3, and some books write there are 4 types. 4 is there. Okay, restriction endonuclease. This 1 and 2, not the 1 and 2, sorry, 1 and 3. This restriction to 1 and 3. This cut the DNA away from restriction site. Cut DNA away from restriction site. Okay, which will generate blunt ends, not sticky ends. But 2 and 4, these are important. These are used in genetic engineering. Okay, and because they cut DNA within restriction site, within the restriction site. Okay, within the restriction site. Within the restriction sequence. Okay, within the restriction sequence. What is the restriction sequence? It is a sequence of 4 to 6 nucleotides, and it is palindromic. What is palindromic? That is, it is equally read from both ends. Okay, like GAA TTC and your CTT-AAG. Okay, this both. This is a palindromic sequence. This is the sequence for EcoR1 action. EcoR1, what it does, that it cuts the sequence from this part and this part. Okay, so I have to generate sticky ends. So sticky ends like this are generated, AATTC and your CTT, AA and G. Okay, sticky ends are generated. So nucleases are there. So again we use DNA ligase. DNA ligase acts as a molecular glue, but it requires energy for its action. Okay, then we require polymerases. Okay, we require alkaline phosphatases as well as the reverse transcriptases. Okay, understood. Talking about vectors, vectors may be like plasmid. Okay, plasmid is a vector. Plasmid is a double-stranded extra chromosomal genetic material. Okay, for example, you have POC. Okay, or you have PBR322. PBR322 is plasmid of Bolivar Rodriguez. Okay, and this is a structure of PBR322. Okay, So you have an AMP gene, ampicillin resistance gene, okay, and you have an tetracycline TETR, tetracycline resistance gene, okay, and you have an ORI region here, okay. This is the site of action for PBU2 restriction enzyme. BAM H1 cuts within the tetracycline resistance gene, okay. ECO R1 cuts here, at least around here. It is the site of action for ECO R1. This is important, ECO R1 restriction site, okay. And this is for your PST1. The PST1 cuts here, okay. So these are the the structure of your gross structure of your PBR 322. I am not going into much details for recombinant DNA technology, as you don't need to retain in this chapter, okay. So plasmid is done. Plasmid will insert less than 10 kb of your DNA. Then you have bacteriophage. Bacteriophage will insert 10 to 20 kb of DNA, okay. Then you have cosmid is 35 to 50 kb, okay. YAC, yeast artificial chromosome, 1 MB, around 1 MB, okay. Then BAC, bacterial artificial chromosome, less than 300 kb, okay. And another MAC is there, okay, that will insert 1 MB. This YAC and MAC 1 MB, BAC is less than 300, okay. Host, what are host? Generally prokaryotic hosts are preferred. Because due to faster rate of replication, okay, faster rate of replication, like your E. coli, your Bacillus subtilis, okay. And in eukaryotic host also lower organisms are preferred, like your fungi, okay, or plant cells. This also can be eukaryotic host. These are prokaryotic hosts are preferred. Understood? Then we have gene transfer methods. Gene transfer we have physical methods, okay. We have physical methods, we have chemical methods, and we have biological methods also, okay. Physical methods, like we have electroporation, like electric shock is given to create the pores in the cell wall, okay, and by this gene transfer can occur easily, electroporation. Then biolistics, like a gene gun is used. Gene gun has helium gas, so you just put helium gas here, inject helium gas here, okay. You have a plastic disc here, and you have the DNA coated gold particles and the screen, the target plant cell put in the screen, okay. So if you put the helium gas, then the plastic disc will fall down along with the DNA coated the gold particles, okay. This will fall upon the cells and the gene will get transferred, okay. This is biolistic, the gene gun, okay. You have opto injections, or you have ultrasonography, ultrasound assisted gene transfer, or you have magnetofaction, okay. This is all. Chemical methods. In chemical methods we have calcium phosphate mediated method, okay, then we have liposome method. Liposomes are injected, okay. Nanoparticles, using nanoparticles, okay. And quantum dot method, many methods are there, quantum dots. Then biological method, we have transduction method. Transduction is with using viral vectors, okay. Adenovirus mediated, so you take the help of viruses and all, mediated, okay. Then exosomes method, okay. Then retroviral and lentiviral, okay. This all modes of transmission of gene transfer methods are there, okay. So let's talk about the applications of molecular biology or genetic engineering, okay. So applications of recombinant DNA technology, okay. So applications of recombinant DNA technology, okay. So first application is the production of drugs, okay. Production of therapeutic agents or drugs, okay. Drugs like your insulin and your hepatitis B vaccine, Okay, and your DNA vaccines. Understood. So these drugs are produced by recombinant DNA technology. Okay, like two plasmids are taken, two plasmids having I region, O region, and I region, P region, O region, such IPO. Okay, these genes are there, and here A gene is there, here B gene is there. Okay, this both are transformed to E. coli. This both are transformed to E. coli, and transformed into E. coli with recombinant DNA vector. Okay, and based on their antibiotic, based on their antibiotic resistance. Okay, they are the fusion products are formed. Okay, the fusion products are formed, like this A is attached to the host genome, this B is incorporated into your host genome. Okay, so this is beta gal insulin A, fusion protein production will occur, fusion protein. Okay, then this will produce beta gal insulin B fusion protein. Okay, and this both fusion proteins are then purified, and beta gal is removed, minus beta gal. Okay, so you get the A chain and you get the B chain. A chain will have about 21 amino acids. A chain will have only 21 amino acids. B chain will have 30 amino acids. So insulin is a hormone of your 51 amino acids. Okay, so A chain and B chain both are combined by disulfide bonds to produce the functional insulin. Okay, such like the hepatitis B vaccine. How can you create the hepatitis B vaccine? So you can introduce a gene called as a hepatitis B antigen producing gene. Hepatitis B antigen producing gene. Okay, and then you can take a plasmid. Then you can incorporate. Okay, this you can incorporate here. Okay, and you can introduce it into yeast. Okay, it is usually introduced into yeast for the hepatitis B vaccine. Okay, and the yeast will produce hepatitis B antibody. Okay, which will be used in the vaccine. Understood? Okay. Then after production of therapeutic agents, we can there is the production of transgenic erythropoietin. You know erythropoietin is a hormone. Okay, which is used to increase the RBC rate production, which is used in treatment of anemia. Okay, you can use make filgrastim. What is filgrastim? Or filgrastim? Filgrastim is basically a substance which is used in a treatment of neutropenia. This is also produced transgenically. Okay, then factor 8 and your 9th. These are also produced transgenically. Okay, and your interferons are produced transgenically. Interferons, your interleukins. Okay, then your epidermal growth factors, okay, which are used in the treatment of burns. Okay, if you suffer from burns, severe burns, then epidermal growth factors are used for your treatment. Okay, then a tissue plasminogen activator, the tissue plasminogen activators. Okay, tissue plasminogen activators. For example, the 3 tissue plasminogen activators: your alteplase, your reteplase, and your tenecteplase. Okay, these are thrombolytic agents. Okay, then the anticoagulant like lepirudin. Histamine is the natural anticoagulant, but you can produce lepirudin, which is also an anticoagulant produced transgenically or by recombinant DNA technology. Recombinant DNA technology is used in human genome project. Okay, used in gene therapy. Many applications are there. Gene therapy is used in your gene fingerprinting. Okay, No, sorry, DNA fingerprinting, not the gene fingerprinting. The DNA fingerprinting, okay. And cDNA library construction, complementary DNA library, okay. Production of transgenic animals. Some transgenic animals are produced. Transgenic animals, okay. Transgenic animals like your mice are produced, knockout mice, like your knockout mice, okay. And your dolly ship who was cloned in 1996. In dolly ship, its parents was an adult fin dorset ewe, okay, who was the nucleus donor, okay. Adult fin dorset ewe, who was the nucleus donor. The nucleus is removed from the cells of the mammary gland, okay. And the Scottish blackface eves, okay, who was the egg cell donor, okay, egg cell donors. So the egg cell is removed from the Scottish blackfaced ewe, okay. The egg cell is then nucleus is removed, okay, nucleus is removed, and this nucleus is grown and incorporate within it, okay, incorporate and given a pulse, electric pulse. After given electric pulse, there is fertilization is induced, and after induction of fertilization, the cell division will occur, and the embryo is implanted. The embryo is implanted again in the Scottish blackface ewe, so it also acts as a surrogate mother, and thus from the Scottish blackface ewe the dolly ship was produced, okay. It's a fin dorset lamb or dolly, okay. So these were the applications of your recombinant DNA technology, okay. Next, related to the molecular biology, is another biochemical technology which is called as the PCR, the polymerase chain reaction, So PCR is a technique which is used for is used as an amplification technique, okay. Polymerase chain reaction is an amplification technique where a DNA polymerase is used, obtained from your tag DNA polymerase is used, okay. It proceeds in exponential manner to produce the desired product, okay. Like you have a DNA, so you have to produce the several copies of the DNA, so use PCR. So it is an amplification technique. What are the requirements of PCR? You need a PCR cycler, or your thermocycler, which is the machine, thermocycler. Then you need template DNA, okay. Then you need DNA primers of about 20 to 30 nucleotides, which act as DNA probes, okay. These are complementary to the target DNA, okay. Primers may be two types, a forward primer and reverse primer. I am not going to details, okay. This primer start the extension of DNA, okay. They start the extension of DNA, okay. Then we have tag DNA polymerase, okay, which is obtained from the Thermus aquaticus, which is a thermophilic bacilli, okay. Then you can use DNTPs, like DATP, DGP, okay, DCTP and DTTP, okay. Let's not go into details and write the steps, okay. The steps of the PCR, okay. What are the steps of your PCR? So first of all, you have the DNA strand here, okay. So first you do denaturation. Denaturation is done at 95 degrees Celsius, okay. 15 seconds to 2 minutes it is done. Denaturation up to 2 minutes it is done. Denaturation is done, due to which the DNA strands will separate, okay. On separation of DNA strands, you add the DNA primers, okay. You add the primer, you have the primer, and addition of primer is called as annealing. It is done at 56 degrees Celsius, okay, for 30 seconds to 2 minutes, for 30 seconds to 2 minutes. Understood? Then afterwards you add the primer. So what happens, the primer is complementary to the target DNA ends, so it get attached like this. The primers get attached like this, okay, primers get attached like this. Then you add your tag DNA polymerase, okay. The process, this is the one, denaturation, the annealing 2. The third is the extension. Extension is mediated by a tag DNA polymerase, or tag DNA polymerase. So tag DNA polymerase attaches to here, okay, and the tag DNA polymerase attaches to here, okay. So here is the attachment of the tag DNA polymerase in both direction, and they catalyze the formation of more and more strands, okay, 2 more strands, okay, production of 2 DNA molecules. And the extension is done from 30 seconds to 2 minutes around, okay, 30 seconds to 2 minutes. So for one cycle we require about 3 to 5 minutes, okay. So for 25 to 30 cycles will require about 3 to 6 hours, okay, 3 to 6 hours. Understood? So you can repeat these steps again to produce more DNA molecules for amplification, okay. So what is the use? That depending upon the time taken, depending upon time, you can estimate the viral load, okay. You can estimate the viral load depending upon the time taken for your cycles, okay. So what are the applications of PCR? Application is that it is first is used for viral infections like your HIV, your COVID, okay, for all the SARS-CoV-2, okay, detection of tuberculosis detection, cancer detection, okay, some prenatal diseases like sickle cell disease, thalassemia, okay, etc., and used in gene library, and it has also forensic applications, okay, like your paternity testing and all, okay. How it is used in the COVID detection of SARS-CoV-2. So SARS-CoV-2 is an RNA virus, okay. So first of all, swab is collected. It gets transported via your VTM, your viral transport medium to the lab, okay. Here in the lab, first step is done, that is RNA extraction. RNA is extracted, okay. This extra step is done in which the RNA is acted upon by the reverse transcriptase, okay, to form the cDNA formation, okay. The cDNA is formed, and this cDNA, the 3 steps are done, that is denaturation, annealing, and extension, okay. Denaturation, annealing, extension. Denaturation at 95 degrees Celsius, 15 seconds to 2 minutes, annealing at 56, 30 seconds to 2 minutes, and extension I have not mentioned the temperature, it is 72 degrees Celsius, okay, 72 degrees Celsius. So this for 30 minutes to 2 seconds, okay. This 3 steps are done, okay, denaturation, annealing, and extension. And with this three steps, again you can estimate depending upon number of cycles, okay. Like if cycles are more, the viral load is less, okay. Depending on number of cycles, you can estimate the viral load, okay. Then we have an improvisation of this PCR. It has an improvisation which is called as your real-time PCR, RT-PCR. RT-PCR was a common term during the COVID times, okay. Must have seen people checking on their RT-PCR reports, okay. This is called as real-time PCR. In this RT-PCR, different type of proofs is used, which is called as Tachman proofs, Tachman proofs, okay. Tachman proofs, and a dye is used, which is called as SYBR dye, SYBR dye, okay, which is a fluorescent dye, okay, and it is detected by your fluorometer, okay. So what is happening in this method is that, let's say here is a DNA polymerase here, okay, the DNA polymerase, okay. So in the probe, this is the probe, okay, this is the probe. In the probe you just label the probe with Q and R, okay. What is Q and R? This is the quencher, of fluorescence, and this is the reporter of fluorescence, okay. This is the DNA polymerase. So what happens is that when Q and R are met together, okay, there will be no fluorescence, okay. But when DNA synthesis is occurring, is occurring, so Q and R are separated due to exonuclease activity, and this separation of Q and R will cause the fluorescence. They will start to glow, okay, and you can measure this amount glow in your instrument called as your fluorometer, okay. So here we complete our RT-PCR, okay. So this was an improvisation, the real-time PCR. So that's all in molecular biology, okay. We learnt a lot, okay. Many things are there in molecular biology, okay, about the DNA replication, okay, what are the changes in prokaryotes and eukaryotes, okay. Then what are the different primates, then P alpha, pCNA, then leading strand DNA epsilon, lagging strand DNA polymerase delta, okay. RNA is H1, FEN, DNA ligase, this all proteins are used, okay. Should be able to remember this. Then types of DNA damage and repair mechanisms, which we discussed: mismatch repair and the faulty mismatch repair, that is hereditary non-polyposis cancer, colon cancer. Base excision repair, okay, and then we discussed the nucleotide excision repair. The faulty nucleotide excision leads to xeroderma pigmentosum. Then double strand break repair, faulty double strand will lead to ataxia telangiectasia, Fanconi's anemia and Bloom syndrome, okay. Then direct repair we discussed. About telomeres we discussed. Then transcription in our prokaryotes. What are the steps in transcription of prokaryotes? Open complex, elongation, termination, intrinsic or row dependent, okay. The clinical is rifampicin, dactinomycin. In eukaryotes, start site is called as fidelity +1 TSS, okay. And RNA polymerase are of three types. Factors are A, B, C's: activator, basal transcription factor, and co-regulators, okay. Then what are the how the DNA is getting bent, okay. The TAF, transcription activating factor, it bends the DNA and forms the pre-initiation complex. Sequences TPP, TF2D, TF2A, TF2B, TF2F, TF2E, and TF2H, okay. TF2H has helicase, nucleotide excision, and it phosphorylates the carboxy terminal of RNA polymerase, activating it, okay. Then termination is by CPSF and CSF, which stimulate the CF, okay, which causes the dissociation of all the things. Then post-transcriptional modifications, we discussed about 5' capping, poly-A-tailing, and splicing, and mRNA editing, okay. Splicing is mediated, which requires snRNA, and 60 proteins are required, okay. 60 proteins are required. And then we talked about translation procedure in prokaryotes, okay, in the 70s ribosomes, and these factors were required: 1 to 6, 1 to 3, and T and G. The charging is there, initiation is there by IF6, then formation of ternary complex is there, formation of 30s initiation complex is there, elongation by EFTU and EFG. EFG provides energy. Termination is by release factor, which is analogous to your tRNA, okay. Then the prokaryotic inhibitors are tetracycline, chloramphenicol, erythromycin, and aminoglycosides. Okay. Then eukaryotic translation we discussed. Activation is there, initiation is there, but there is first mRNA activation in which 4F is attached. Okay, and your 4B and PAB are also attached. 4F, 4B, and your PAB, these three are attached. Okay, in prokaryotes signed Delgarno sequence is there, but in eukaryotes Kozak consensus sequence is there. Ribosomal dissection dissociation is will occur in the presence of EIF6. Okay, then ternary complex will get formed. Then 43S pre-initiation complex, 48S initiation complex will form, and 80S initiation complex will form. Elongation will occur by EEF1, which provide energy for tRNA attachment to A site, and EEF2, which provide energy for peptide bond formation. EF2 is inactivated by a diphtheria toxin. What are the inhibitors of protein synthesis? Diphtheria toxin, cycloheximide, and ricin. Puromycin is the inhibitor of both. Post-translational modification, we discussed proteolysis and covalent. Proteolysis is removal of initiator methionine, pro-protein activation, cleavage of signal peptide. And covalent, we have addition of carbs, prosthetic group addition, phosphorylation, dephosphorylation, disulfide bond formation, OH group addition, acetylation, isoprenylation, gamma carboxylation. Okay. Then discuss some about gene control of gene expression. Okay, in prokaryotes and eukaryotes. About the lactose operon we discussed. Okay, what are the effects on in the presence of lactose and absence of lactose. Then we discussed about the tryptophan operon also. Okay, which mechanism is attenuation, and the bacterial molecule switching in a bacteriophage lambda I told a little bit. Okay, then control of gene expression in eukaryotes. In DNA level it is epigenetic modification are there. Acetylation lysine acetyl, methylation by histone. Okay, at transcription level, promoter, enhancer, transcription factors, and DNA binding motifs, these are the things which will regulate it. At post transcriptional level, splicing and mRNA editing are then are your mRNA stability. This is not your mRNA stability. Okay, we discussed this all. And then translational level also, EIF2 and EIF4E, these two are causing the regulation. Okay, then we discuss about recombinant DNA technology, which is also a part of this chapter. Okay, what are the steps of recombinant DNA technology? What are the enzymes, nucleases like ligases, polymerases, alkaline phosphatases, reverse transcriptases? What are the vectors like plasmid, bacteriophage, cosmid, YAC, BSC, MSC? Okay, and discuss the structure of EcoR1. Okay, then about your host, gene transfer methods, physical, chemical, biological. We talked about the applications, how the insulin is produced, and other factors, other things that are produced by rDNA. Okay, we discussed how the dolicip was cloned, although it's not a part of our syllabus. Okay, then PCR, about the PCR, what are the the requirements of PCR? Okay, what are the steps? That denaturation, the annealing, and the extension. Denaturation is at 95, 15 seconds to 2 minutes. Annealing is at 56, 30 seconds to 2 minutes, and your extension is 72, 30 seconds to 2 minutes. Okay, depending upon the time you can estimate the viral load. Okay, applications we saw, and about the real-time PCR we discussed, in which the quencher and the reporter, if they are together, they will not produce, but if they are separated due to exonuclease activity of DNA polymerase, then fluorescence will be observed. Okay, so this completes the summary of your recombinant of your molecular biology, and that's all molecular biology. I hope it all made sense to you, and you understood all the concepts clearly. Okay, this much concepts are only there. I tried my level best to cover each and everything, the inhibitors and all. So you can write the long questions, many important long questions like your translation, transcription I have covered already. Okay, so that's all in today's video. If you like the video, do like, share and subscribe, and check out the other videos in this series as we progress on. I have already covered the big chapters like neurophysiology or hematology, and the other chapters like your GIT in complete details. So this is a revision of molecular biology also, and now it is finished. Okay, so that's all for today. Let's meet the next time. Thank you and have a nice day.